Did edit this after too? I think it's all
So you'll be posting these lectures online.
okay. Try it.
You're gonna try it.
Okay, thanks, Louis. We'll figure out. Yeah.
Depends how successful they are, right?
We're going to get going in 1 second. Actually, there's an announcement before we get going by the UPSA co-president this year about test packages and things related to your midterm. If you want to make your announcements in.
All right, sure.
Hey guys, so if you're interested in purchasing a PSL300 test package, Upsell will be selling them for $5 at the end of your class on Thursday. So we'll be setting up at around 7 o'clock.
You guys sent information very bit, right?
So yeah, we'll be there around 7 o'clock. So by the time you guys are done, everything should be set up. So again, there's still PSL300 test packages for $5 at the end of Thursday. Alright? Best of luck, guys.
Okay, we're going to try things a little different. For those of you who haven't noticed, I'm not Dr. Key. We kind of look the same, but we're not related in any way. We're going to try things a little bit differently. I'm going to try to stream this lecture and capture it for you on YouTube and make the recording links available for you after class. Especially with the neuroanatomy section that's coming up, it's important that you stay on top of the material, and wherever possible, I'm going to try to make sure that all the material is available for you in a timely way. If you have any questions, by the way, I do have office hours. I know your midterm is next week. I'd be more than happy to talk to you, especially this week before the weekend gets started. The best way for you to do well is really to stay on top of the material, and today you're going to be hit with a lot of material. So again, this section is really difficult to actually study for. You should study as efficiently as possible wherever you can. Concentrate on the lecture notes. I'll be pointing out things to you during the lecture that you should be paying a little bit more attention to. I will also be engaging you as much as possible online, and so it's really up to you. To do well, we'll give you as many resources as we possibly can, whether it's a YouTube recording or whether it's a lecture recording, online sessions, but it's really up to you to do well by staying on top of the material and coming to office hours if you don't understand any of the material. So I'm just going to give you an overview. You don't have to start worrying about any of the notes until I tell you to start worrying about what we're going to be talking about. So we're going to be looking at some really difficult topics in the next couple of lectures. So we're going to start off today looking at the general properties of the sensory system, and then from the sensory system talking about the neuroanatomical pathways that lead up to the brain. And that's not easy. There's a lot of anatomy involved, there's a lot of physiology involved, and you need, again, to stay on top of things. So in terms of neurophysiology today, I want you to understand why and how we interact with the outside world around us. And especially over the next couple of lectures, I'm going to talk to you about the sensory motor system. So it's one thing to see things, and it's one thing to process smells and other senses, but we need to understand how that works. How does your brain actually relay a signal from the periphery out here to your brain, and how does your brain put all of that information together? And along the way, we'll talk about these specialized senses, and we'll look at how these different specialized senses actually come to pass. Whether you think so or not, you're actually a series of pre-programmed reflexes. You have adapted your reflexes over time. You don't have the same reflexes that you were born with, and it's very likely that you'll lose some of your reflexes before you die. But all of that is stuff that we're going to be talking about, and especially in the next little while, how these pre-programmed reflexes make up things like walking, how it makes up things like breathing, and why your nervous system is capable of doing all of these things throughout your entire lifespan. So we're going to talk about a couple of common themes throughout these lectures, and I want you, wherever possible, that is a great ringtone if it's a ringtone. If you're watching a movie, perhaps watch it a little bit later on. Thanks. So we're going to talk about some of the generalized concepts in neurophysiology. You've had a basic overview with Dr. Key. We're going to go through the basic plan of the nervous system. The brain is a really complex structure. The things that make up the brain, the nerves that you looked at last week, are really complex structures. But I want you to start looking at it as sort of a really intricate system that has a basic plan that you can easily understand. We're going to talk today about how signals get into the brain. And specifically, we're going to be spending some time looking at what's known as the somatosensory system. How does your skin tell you what's going on in the outside world around it? In addition to that, starting on Thursday, we'll be looking at special senses like vision, and we'll be looking at how all of these signals get into the brain and how getting those signals into the brain allow you to react to what's going on around you. So it's one thing to touch something and then to realize that you shouldn't be touching it and withdraw your hand. You're able to do that simply because you have reflexes, or you're able to control volitionally by your own free will the movement that you've been able to generate. And we'll be talking about the outflow in the upcoming weeks as well, and a little bit starting today. And then we'll talk about specialized outflow systems, the autonomic nervous system, something we don't really take into account every single day, but that are so important for some of the basic processes that keep you alive, that keep you healthy, and keep you active. So again, you don't have to worry about all of these different things on this particular slide. I'll tell you when you should start concentrating on things for your midterm. It's just to give you an idea of the types of things that we're going to be looking at, and to give you some terminology that I'll be talking to you about later on today. So in particular, we're going to be looking at the central nervous system. The central nervous system is going to process all of that information that I've been talking to you about, whether it's a chemical system or a chemical sense, or whether it's a somatosensory sense. All of that is going to be processed in the central nervous system in very specific areas of the brain, and we'll start looking at that today. All of that information, by the way, has to be relayed through the peripheral nervous system, and this is where we're going to start our conversation in a few slides. And again, we won't be talking about the outflow, how your body responds, until much later on in the course. So the most important thing for you to take away from this slide is that we have two different information pathways, one going into the brain, and that is an afferent system. And that afferent system makes use of your spinal cord. It has pathways in your spinal cord that lead up to your brain. And then we have this efferent system that comes down from your brain to interact with different muscle systems that make up reflexes, volitional movement, etc. And we'll be talking about both of those today. So afferent meaning going toward the central nervous system, or CNS, versus efferent, which is coming out of or away from the CNS. So again, you don't have to worry about any of these numbers. It may sound like an inordinate amount of neurons that we're talking about. We have somewhere, and we're not able to count. It's a best guess. It's a best guess of an adult brain, but we have no way to know for sure if you really do have 100 billion neurons. Right now, that's our best guess. There are some individuals that say you have about 60 billion neurons, others that say that you have about 100 billion neurons. We're not really sure. We know that we have a lot of different neurons that comprise your CNS, and that most of them reside within the brain. And we'll be talking about that in some more detail today, specific groups of neurons in the brain today, and then all of these, of course, making up these connections between other neurons known as synapses. And you guys talked about synapses with Dr. Key, is that correct? So you guys are all experts on synapses, correct? Not so confident now. Okay, that's okay. So we won't be spending a lot of time on these synapses, but they're really important in terms of communicating across these billions of neurons. We have trillions to quadrillions of synapses, and these will become really important as we go on in today's lecture. So at this point, if it's not clear to you, this is something that you might want to start paying a little bit more attention to. There are a lot of different definitions for what comprises the CNS. I'm going to keep it really simple. The CNS is really your spinal cord and your brain. In other words, it's everything that's covered by a bony structure. So your brain is covered by the cranial vault, and your spinal cord is covered by the vertebrae, and we'll be talking about that a little bit more. some of those send out projections. Are they coming from the CNS? Yes, they are, but they're leading away from the CNS. They're part of that efferent system, and these will be comprised of the peripheral nervous system. And these peripheral nervous system neurons, which I'll abbreviate as PNS, are all of the axons as well as the cell bodies or somata that are not protected by the bone, nor are they protected by the blood-brain barrier, because the blood-brain barrier is another defining feature of what makes up the CNS. The PNS is exposed to whatever else is going on in your environment, in the extracellular fluid. The CNS, the brain, the spinal cord is a very protected structure, and I think Dr. Key went through that with you in the last lecture as well. This is also a really interesting thing. The most heavily protected part of your nervous system is actually the most vulnerable. So if you have a stroke, if you damage a central nervous system neuron, if you have a lesion in your brain or in your spinal cord, the most protected parts of the nervous system are actually the most vulnerable. They don't grow back. Once you've lost it, it's gone forever. And those of you who have know individuals who have damage to their spinal cords, who've had stroke, know that it's incredibly difficult to recover function because that part of the nervous system doesn't grow back. The part that's not well protected, the part that makes up the peripheral nervous system, the axons and the cell bodies that sit outside of those bony structures, not protected by the blood-brain barrier, in fact, you can damage those. You can twist those, you can actually sever them, and they will grow back. And this is, at least for vertebrates, and the reason why I have this goldfish up here is that in certain vertebrate species like the goldfish, the CNS does repair, but in humans and most mammals, the CNS does not repair. And we still don't understand what makes the difference between why central nervous system neurons in animals like the goldfish can repair themselves. You can crush the optic nerve, it will grow right back. Whereas in us, if you damage my spinal cord, it will never grow back. And again, that is a huge area of research here at the University of Toronto. I'm not sure if all of this made it onto your slides, so there are some things that I want to point out for you, and these are things that you probably want to start reviewing this weekend as you go back and study for your midterm. So functionally, we define the CNS as all those things that are protected by bone as well as by the blood-brain barrier. We can also identify different types of cells that surround the neurons in either the peripheral or the central nervous system. So for example, the neuroglia, the cells, the support cells that are found within the CNS are very different than the types of cells that are found within the PNS. So we have things like oligodendrocytes, which you learned about in the previous lectures, that make up the myelin that cause nervous system signals to be conducted faster. And we have different things like astrocytes, and we have microglia, as well as these other cell types which are known as ependymal cells. All of these glia are unique and found only in the CNS. These are not found in the peripheral nervous system. In addition to that, these have other functions other than just making things like myelin. For example, they will be marked by specific proteins. So if you have CNPase staining, then you know that you have identified an oligodendrocyte. If you have a tissue that has CNPA staining and therefore oligodendrocyte staining, you know that you're in the central nervous system, regardless of the types of neurons that you find around it. If you found GFAP staining, then you know that you have an astrocyte, and that most likely, because it's an astrocyte, you are in the CNS. And again, the same thing for microglia, which are the immune cells found within the central nervous system. These cells, unlike the astrocytes, tend to have an immune function, and so we can mark them with an immune marker like CD11. The last type, the ependymal cells, are involved in helping to form those tight junctions that make up that blood-brain barrier, as well as eventually, during development, forming what's known as these radial glial cells. And again, a really important cell type that I'll try to get back to later on. All of these are unique to the central nervous system, as opposed to these other neuroglial cells. They are not neurons, they're support cells, like the Schwann cells, that are also capable of producing myelin, but not in the CNS, only in the peripheral nervous system as part of the PNS. Again, like the oligodendrocytes, we have specific markers for these Schwann cells that produce myelin. We can identify them using an antibody known as 1E8. specific only for these Schwann cells, or we can identify them using a marker against a protein found only on the Schwann cells known as HERP. These last cell types are actually the most intriguing type of cell types. The satellite cell is found associated with part of the peripheral nervous system that is known as the sensory ganglia. And we'll start talking about the sensory ganglia today. We're going to be looking at somatosensation, how you feel touch. And around the cell bodies of these sensory ganglia cells, we would find these other specialized glial cells known as satellite cells, which can be stained using glutamine synthetase, abbreviated here for you as GS. Do you have any questions about any of those? So these help us to identify the neuroglial cells that surround neurons, because neurons in the CNS and PNS, really, they don't look very different. And in terms of pathology, to identify where you are, it helps to be able to mark these different support cells that you always find. So you don't have this slide and you don't need to worry too much about this. We'll talk about this later on in the course when we get into rhythmic behavior. So anything that has rhythmicity. Your breathing is rhythmic. You breathe in, you breathe out. Walking is rhythmic. All of these things don't require the higher parts of your brain. It only requires the brain stem. And we'll talk about the role that these glial cells that I just mentioned to you have in that. We know from a paper that was published last week at McGill University that these astrocytes that surround, and these astrocytes are here for you in white, that surround these neurons that are stained in red, found in the brainstem, actually are really important in terms of determining how you breathe in, how you breathe out, how you maintain autonomic functions. And so these glial cells have a much more important role than we thought previously a few years ago. So I'm going to mention a couple of things again in terms of anatomy. It's really to help you get organized in terms of the different structures that I'll start mentioning to you. You don't have to memorize anything here. I'm hoping most of this is a review for you. When we start looking at the spinal cord, and here is one part of the spinal cord, and we'll look at the different sections of the spinal cord, like the dorsal horn and the ventral horn, we will see that information comes in. It's afferent information from the different sensors that are found on the skin. And here is an example of a type of receptor that's found on the skin, sending information back, afferent information, going through the cell body found in the dorsal root ganglion, and it will eventually convey that information into the spinal cord. And all of this, again, is where it gets into the CNS. So previously, that information has been in the periphery, and now this information is getting relayed into the CNS, and that CNS acts as a way to respond to that information. What do you do with that information coming from your skin? And again, that is relayed back out through these efferent fibers, which might innervate things like muscle or might innervate other different structures, as we'll see later on. So within the CNS, and again, it's just for your information. I'm not going to ask you a question on this. I want you to understand what this means. If we have an axon bundle in the CNS, we call it a tract. If we have an axon bundle in the PNS, even though it's exactly the same thing, instead of calling it a tract, we call that group of axons a nerve. Okay, so even though it's exactly the same thing, the same structure, an axon bundle is named differently within the CNS as well as in the PNS. If we have a group of cell bodies together in the CNS, and you'll see this very soon, we'll call this a nucleus. It's a nucleus of different nerve cells. Okay. In the PNS, if we have a whole collection of these cell bodies, we call it a ganglia. So this dorsal root ganglion is really a collection of different nerve cell bodies that sits in the PNS. Again, the myelin-forming glia in the CNS are the oligodendrocytes versus Schwann cells in the PNS. Is that okay for everyone? Feel free, by the way, to interrupt me at any time if you have a question or if there's something that you would like me to clarify for you. Again, this is just for your own information. I hate memorizing numbers, and I'll tell you which numbers you need to memorize, so you don't have to memorize how many thoracic versus how many cervical versus how many lumbar types of segments we have within the spinal cord, but since we're talking about the CNS, it's useful to know that there are different levels of spinal cord segments. So we have ones that control things up here in the upper part of your body, in the cervical and thoracic region, and then we have other groups of neurons that control your legs and lower structures below. So there's an organization, in other words. It makes no sense to have some part of your spinal cord down here controlling things up here at the level of the thoracic spinal cord. It also becomes really important for another reason. We name these according to the different spinal cord segments that we find associated with the vertebra, the bony shell. Remember, CNS, we talk about bone, protection by the bone. So we name all of the different segments of the spinal cord related to the different vertebral sections, or these bony projections that you see here. But the spinal cord actually ends early, before your spinal cord stops way down here. Your spinal cord stops here, and the spinal vertebra, the different bone structures actually go farther down. And so this section down here is nothing but the axons, the axonal projections that we would see in the CNS. So when we do things like a lumbar puncture, which is what LP stands for up here, where would you do it? If you don't, I just told you that your spinal cord is part of your CNS. Your CNS is not capable of regeneration, and you're going to be sticking a needle into someone's back, into an area of the spinal cord, theoretically, possibly damaging the spinal cord. So if you want to avoid damaging the spinal cord, and you know that the spinal cord ends early, where would you think of potentially doing a lumbar puncture? Sorry? After it ends. Okay, so where does it end, do you think? Roughly. Yep, so after the last lumbar segment, at around the sacral end, that's where you would actually do this lumbar puncture. There's no more spinal cord left. It's all these cauda equina, all of these axons, and no cell bodies to damage. Your needle will just push these fibers away. So understanding the anatomy and physiology becomes really important and really interesting when you understand why we talk about the spinal cord in the way that we do. So this slide and the next few slides are ones that I'm going to ask you to circle to make sure that you spend some time and go back to a little bit later on to review some of this material. And I'm going to go through the anatomy of the spinal cord in a couple of different ways. One, I want you to understand that the overall structure of the spinal cord is a well-conserved structure. So in other words, we have this structure here in the middle, in this butterfly shape, which is known as the gray matter, or the central gray matter. And I'll get into that in some more detail on the next slide. And the central gray matter, again, it's really highly organized, has three different regions that I'd like you to be familiar with as well. So at the very top, we have this dorsal horn, and the horn just means it's projecting out toward the surface. So this dorsal horn region of the gray matter is important because this is where you will get sensory signals coming in. So this dorsal horn here on the left tells you that sensory information, afferent information, is coming here through that region of the dorsal horn. So sensory information comes in to the dorsal horn. It doesn't matter what segment of the spinal cord you're looking at, whether it's cervical, whether it's thoracic, or whether it's sacral, even though the shape of that central gray region that you see here changes over time or with distance, that overall it's exactly the same. At the very top in the dorsal region, we always have sensory input coming in. So once that sensory input comes in, it has a choice. It can do lots of different things. One of the things that you'll find in the spinal cord is that in this zone here, which is known as the intermediate zone, and it's highlighted for you on the left in gray, this intermediate zone is where some basic processing of that sensory information will occur. You will see something analogous to this in the brainstem, and that's one of the reasons why it's highlighted for you. So some basic processing through different types of synaptic connections will occur within this intermediate zone, which is why you see these other neurons in white being shown for you here on the right. Eventually, though, we need to interact with the outside environment. We need some sort of efferent output, and this efferent output is in the form of this, usually a motor neuron that will leave through the ventral horn of the spinal cord. So motor neurons are found in this ventral region, and these are the efferent signals that usually cause some sort of motor activity, contraction of a muscle, or something very similar to that. I always ask this question. I'm always surprised by the answers, and I'm hoping that you will not disappoint. Why do the different sections of the spinal cord, as you go from the very bottom, the sacral region, way down here at the tail end of things, all the way up toward the cervical region at the very top, why is there a change in the shape of that central gray? I'm hoping you can all see that there's a change in the central gray, but why do you think there is such a change? Any ideas or any thoughts or any theories as to why? Yes. So more motor neurons. Okay, that's a possibility. That's a good possibility. Maybe the motor neurons down here at the bottom supply bigger groups of muscles. Like your leg muscles are obviously bigger than your arm muscles. Certainly a possibility. You need more gray matter, more motor neurons down there. Wouldn't really tell you much about this. Maybe you have more sensory information coming in. But think about this, right? So that information that comes in from the periphery gets integrated through the dorsal horn. Does it always stop there? No. So I told you at the very outset, your brain has to be involved in the processing of this information. So in other words, that information from my legs at the very bottom, at the sacral end of things, has to send its information all the way up to the brain, right? And the way in which it does so is through these different funiculi. And these different funiculi are found in this white matter. So we haven't really talked about the white matter, and you can see that the size of the white matter continually increases the farther up you go toward the brain. So up here at the cervical, just below sort of the base of my neck, base of my skull, the cervical region actually has lots and lots of white matter, very, very little gray matter, because all of the axons from the legs and arms also have to find their way up through the cervical region of the spinal cord as well. So there are, again, reasons why the white matter increases in size. More and more axons that are found here in the different funiculi are going to be important. Do you have any questions about that before I go on? No? Okay, so this is, again, another slide that I'll actually ask you to make sure that you are familiar with. Again, a little bit of anatomy for you. When we talk about this ventral horn down here, another way to say ventral horn is the anterior horn. Just like the posterior is the dorsal region, we can use these terms interchangeably. I tend to favor using ventral dorsal, but if you hear me switching back to anterior or posterior, these are what those things mean. So if we go back and look at the shape of the spinal cord, and here it is for you, this white matter that sits all the way out here versus the central gray region and the intermediate zone that's grayed out for you, this white matter region contains all of the myelinated and unmyelinated tracts. So if you're thinking that myelin is white, therefore the white matter that you see surrounding the gray is only white matter because it's myelinated, you're wrong. There are also unmyelinated fibers that go up through those regions as well. The gray matter has a lot of cell bodies. We also find in this gray matter unmyelinated axons as well as neuroglial cells that are found here as well. And again, I've mentioned these different horns, the dorsal and ventral horns, in the previous slide. So now I'm going to talk to you about why we have white matter versus gray matter. So when we look at the different regions and we look at these different sensory signals, I told you they are all going up in toward the brain. So we can look at the white matter being divided into 6 different regions within the spinal cord, or 6 different funiculi within the spinal cord containing all of these tracts, which again is a term that we use in the CNS. So when we look at these tracks, and you don't need to worry about the details here on the right, when we look at these ascending tracks, these are taking information that is coming into the spinal cord and then sending information up into the brain. There are also tracks that have to come down from the brain through the spinal cord down toward these ventral areas, and there are also descending tracks. Together, in total, we have 6 different columns or funiculi that are involved in taking information up or information down from the brain through the spinal cord. And again, don't worry about the details here on the right. Okay, so if we take a look at the information coming down, so you have information from somewhere higher up in the brain and it's coming down to the spinal cord to control different types of activity, these descending motor pathways are really important. And these are really easy to identify as well. These brainstem motor areas that we'll eventually see later on in the course are going to be sending their information down the spinal cord in specific areas. In other words, if I send information from my cortex through the white matter in the spinal cord, we will usually find it here in the lateral region of the spinal cord. So this is where we find the corticospinal tract. Is that clear for everyone? So I'm sending information down. All of these pathways that I'm going to mention to you, reticulospinal, vestibulospinal, corticospinal, and rubrospinal, are all coming from somewhere higher in the brain down to the spinal cord. And we know that it's a descending tract because there's always something in front of the spinal tract. Corticospinal, from the cortex to the spinal cord. Rubrospinal, the rubral nucleus down to the spinal cord, the vestibular nucleus down to the spinal cord, and the reticular formation down to the spinal cord. And all of these have different types of activities. So the corticospinal tract and the rubrospinal tract that you find only descending here on the right, it would also be mirrored, by the way, here on the left as well. It would be found in this region laterally. These are important in things like turning your head, making sure that your head is staying upright. And if you damage these regions, your head would be flopping around a little bit. It also helps you with postural support. So these pathways are all the things that are involved. If you damage these, you aren't going to be able to maintain your posture. You can't maintain your head. You can't do all of the sort of turning of your head, stability, etc., that carries information down through these different tracks. The reticulospinal and the vestibulospinal are also really important. They're important for a couple of reasons. One, as they're coming down from the brainstem, and these two are coming from the brainstem, they're going to project to motor nuclei that are found in these intermediate zone regions. These types of spinal cord tracts are both inhibitory as well as excitatory. The one that I mentioned to you previously are always going to be excitatory. Okay, so the lateral tracts that I mentioned to you here, the corticospinal and rubrospinal, they're always going to be turning things on. But these tracks here, the reticulospinal and vestibulospinal, can either turn things on or turn things off. So that both of these are inhibitory and or excitatory. So eventually, we need to look at some higher level structures, and we need to take a look at some of the other structures within the brainstem. So the brain is really a simple structure if you want to think of it this way. So take a head of lettuce. That head of lettuce represents the cerebral cortex, which I'm going to get to at the very end of things today. So the cerebral cortex is this head of lettuce, and what you do is you take a celery stick and you shove it right into the head of lettuce, and that represents sort of this brainstem region that I'm going to be talking to you about in the next few slides. This celery stick that I just put into this head of lettuce can be further subdivided into this midbrain region at the very top, this pons region, and then this medullary region here at the very end. And this is where the spinal cord would be starting. So if you've been following along, do you think there's a physical break between where the spinal cord stops and where the brainstem begins? Like it just stops, dead end of the road, there's a gap, and then the brainstem starts. Yes? How many of you think yes? There's a physical break in the spinal cord, and then the medulla starts like after a little break or a gap? No. How about it's all continuous? It's just like one big tract. Okay, so You're 100% correct. There is no difference between where the medulla starts and where the spinal cord starts. It's all one continuous tract. And so these are artificial sort of designations. So again, when we start looking at this structure that we see here at the very top of that celery stick, this diencephalon is also a really important structure that we'll start to see today. This is really important for two reasons. The thalamus that we see as part of the diencephalon is a relay station. And if you don't remember anything else from today, I want you to remember that all of the different nuclei that we'll be talking about in the thalamus, they all act as relay stations between the spinal cord, the brainstem, and this relay up toward the brain. There's this other structure that's found in this diencephalon known as the hypothalamus. And this hypothalamus, some of you will know from other lectures or from other courses. Why is the hypothalamus so important? You can't actually survive with the hypothalamus. You might be able to survive maybe a week, but if you don't have a hypothalamus, you're not going to be able to survive very long. Why is it so important? Yes? Memory? It does flavor memory. It does contextualize memory, and we'll get into the limbic system later. What else does it do? Yes? So this is really important for homeostatic control. It's also that interface between the nervous system and the endocrine system that you'll be learning after you finish this module on the CNS. So if we go back and we start looking only at this structure here, so everything from sort of the pituitary gland up toward the hypothalamus and all of the structures in the brainstem, and we leave on the cerebellum and we remove all of the cortex, and we'll get to the cortex at the very end. If we were to take all of that away, we would be left with this. And why is this structure so important? I told you that the brainstem is really important because it gives you autorhythmicity. It helps you to organize how you move, how you breathe in and breathe out. And many of you will know about the brainstem for autonomic functions or autorhythmic functions as we mentioned them earlier on. So I'm going to point out a couple of things to you here as well. This gray region that you see outlined for you that's running all the way through the different sections of the brainstem, whether it's the pons or whether it is the medulla down here at the bottom, this gray region is exactly the same as the intermediate zone. Do you guys remember what the intermediate zone in the spinal cord did? So it helped to integrate signals and sort of sent another signal out. This intermediate zone has an analog. It's a continuation into the CNS, and it's what's known as the reticular formation. And this loose network of cells, so cell bodies and different axons, just like in the spinal cord, is found in this central region, just like the intermediate zone was found in the middle region of the spinal cord. And this reticular formation helps to organize all sorts of different behaviors, and we'll be seeing how these different behaviors are organized a little bit later on in the course. For the time being, I'm going to ask you to know that this intermediate zone is a continuation or continued by this reticular formation, which is a loose connection of different neuronal cell bodies as well as axons that help to integrate different types of signals in the brainstem instead of in the spinal cord. If we were to take a look at different regions of the brainstem, so we were to take a slice through the brainstem region here at A, and then we took a look at the section, we turned it around, we turned it perpendicularly, and then we looked at it face on, and we looked at what we saw in the midbrain region, or we took another slice lower down in the brainstem region and we looked at the medulla, we'd see a couple of things. And again, you don't need to worry about all of the details that are found on this slide right now. That will come a little bit later on in the course, so you don't need to worry about things like the periaqueductal gray or the different motor nuclei. We'll talk about those later. But one of the things that you see is as you go further in the different areas of the brainstem, we will see that these central gray regions, this reticular formation, starts to look a little bit different. And again, there are lots of different nuclei associated with it, but there are going to be some structural shifts. Because we need this intermediate zone, some of those neurons that we normally find in the dorsal region of the spinal cord in the brainstem get shifted around, okay? So again, I'm going to talk about that in the next slide. The most important things for you to take away is that this interconnected loose association of axons and neuronal cell bodies in this reticular formation, which represents the middle or intermediate zone in the spinal cord, because it's changing its shape as you go along the brainstem, means that other neurons that sit around it have to also change their location. And I'll give you a good example of that in a few moments. Or actually, this is actually not a bad place to talk about it. Do you remember in the spinal cord, the ventral horn, what kind of information or what kind of neurons did we find in the ventral region of the spinal cord? Was it sensory information or was it motor information? It was motor. And so instead of finding it here in the ventral region, some of these motor nuclei are going to start to shift, which is why this slide is called these structural shifts. This ocular motor group of neurons, for example, is also not found in the ventral region. It's also shifted in its position as well. And some of the sensory nuclei that you might associate with sensory information, these sensory nuclei are also going to move slightly as well. And really the only thing to take away from this is that because of this reticular formation changing its shape and position, some of the other nuclei have to change their position as well. So sensory information is not always found in the dorsal region as we would in the spinal cord. It can be shifted around, and the same for the motor neurons that we would find in the ventral region. They also shift around as well. Do you have any questions about this before I go on? I'm trying not to concentrate too much on the details on this slide right now. We'll come back to that a little bit later on in the course. Okay, so the last section that I'll sort of talk about is the medulla, and we'll start looking at the medulla in a little bit more detail. So this is the section between the brainstem and the spinal cord, and it connects all of that information from the cortex all the way down to the spinal cord. It has many of the reflex centers that I'm going to be talking to you about, has many of the different relay stations. And all the things that you would find in here are things like the autonomic nuclei. So if you're controlling how your gut moves without you consciously being aware of it, it controls the visceral functions. Your internal functions are found at the level of the medulla. The sensory and motor nuclei of 5 of the 12 cranial nerves are found in the medulla as well. So in other words, there's an overrepresentation of the sensory and motor nuclei of the cranial nerves. And again, it acts as this relay station for both sensory and motor pathways. And again, it's a little easier to see this on this particular slide, and you have to remember this central region down here is that reticular formation, which in the spinal cord, when we looked at it, would be that intermediate zone. And since the brainstem is a continuation of the spinal cord, it would make sense that all the structures that are associated with the medulla would be exactly the same, but yet they're not. They've been shifted around a little bit, and we can see that the most in this particular slide. So for example, this nerve down here, this cranial nerve, hypoglossal nerve, cranial nerve number 12, I like making I want to find out a little bit more about this class. So if you can tell me what the cranial nerve number 12, those of you who have not taken Anatomy 300, what does that control? You can either tell me or show me. Yeah. Any ideas? Yes. Yes, it controls your tongue. So I will give you one of your multiple choice test questions in advance of the test on Thursday. Good job. So this motor nucleus here, cranial nerve number 12, it's a motor neuron. It controls your tongue. It controls how you stick out your tongue and all of that other good stuff. Is not found here in the ventral surface. It's actually found up here in the dorsal surface. It's moved, in other words, because of the shift in the intermediate zone. And again, some of the sensory nerves are also not found in the dorsal region. They've been pushed down to the lateral regions. And again, you don't need to worry about all of the details that are associated with this. So the motor nuclei are not where they're supposed to be. Some of them are found in the dorsal medial region. Some of them have been pushed off to other regions as well. Eventually, what we want to do is look at how do we actually process all of this information. whenever we're looking at these different systems, we always want to be looking at how this information gets into a particular region of the brain. In order to do that, we need to understand that the brain is really not a homogeneous structure. There are segments that are more tightly associated with one type of information processing than others. Many of you will know, for example, that the frontal lobe has a variety of different functions. From our standpoint, we're going to be looking at it really as a region of the brain that's involved in controlling voluntary motor movement and activity. And yes, those of you who have taken psychology, you're 100% correct. It's involved in decision making. It's involved in many complex behaviors. But for physiology, we're just going to be looking at it in terms of making these complex voluntary movements. This parietal lobe region that you see here is involved in starting the process of disseminating as well as decoding things like touch, as well as other somatosensory types of information, including things like pain. And we'll be talking about this region a lot in the second half of today's lecture. The occipital lobe, which is where we're going to be spending time on Thursday, is where we get conscious visual perception. And again, this area of the brain is tightly associated with that. We wouldn't find information, for example, about somatosensation. We would find it more likely out here in the parietal lobe. And this temporal lobe that you find on the sides, this temporal lobe region that you find here, is where you start to develop an appreciation for some of the other sensory systems, like the auditory system, as well as the sense of smell. All of that gets processed within the temporal lobe region. So your brain has a number of different regions that are involved in decoding, deciphering, as well as processing different types of information. Some are really good at touch, some are really good at vision, some are really good at smell, but they do have localization, and there is a structure to these 100 billion neurons. The brain, by the way, for those of you who are really interested, and I highly recommend it, I like sending students out to go and do some shadowing. If you have the opportunity this summer and you'd like to go watch a neurosurgical procedure, let me know. I'll try to set you up with that. The brain isn't color-coded. So when your neurosurgeon drills in and opens up the skull and they take a look, it's not color-coded like this, right? So how do we know like what part of the brain is the frontal lobe, and how do we know what part of the brain is the temporal lobe? Where does one stop and where does the other one begin? A lot of that is related to these different invaginations, which are known as sulci, and we typically orient ourselves around the central sulcus, and it looks fantastic here, right? It looks so easy to identify. If you ever see a real brain, I would guess that 98% of you would not be able to identify where the central sulcus is. I have a hard time identifying where the central sulcus is, and I've been looking at brains for a number of years. And it's not so easy to identify, but this is still the way in which we classify things. So the central sulcus is one way to divide up the frontal lobe from the parietal lobe, and then we have this lateral sulcus, which helps to divide the frontal lobe from the temporal lobe. And again, there are other fissures, and there are other types of invaginations that allow you to make a demarcation of the occipital lobe, and I'll talk about that a little bit later on. If I were to take any one part of the cortex, bless you, if I were to take any one part of the cortex, doesn't matter if it's frontal lobe, parietal lobe, or anywhere else, anywhere in that cortical structure, we would see that the overall organization would look something like this. And I want you to be aware of this as we start thinking about how the brain processes things throughout the rest of these lectures. And this is a slide that I would recommend that you spend some time going over if this is brand new for you. If this is the cortex, on any one part of the cortex, occipital lobe, temporal lobe, frontal lobe, wherever we're looking, we see these 6 different layers. And these 6 different layers actually have different functions. There is an organization, in other words, to the neurons that we find in these different layers. So for example, oftentimes if we find neurons that are found in this cortical layer number 4, we would see that these are typically identified as these large pyramidal cells. And these pyramidal cells are identified by the shape of their cell body. And you can see that they're sort of triangular. And these cortical neurons that we find here sort of between layer 3 and layer 4 are really going out to other regions of the cortex. And every neuron that's found between layer 3 and 4 would send their axons out to other parts of the cortex. And again, all of this is where the cell bodies and some of these axons are. And some of these, for example, in layer 6, would be sending their projections not to other regions of the cortex. We would trace their axons all the way out to the thalamus, another area not part of the cortex, but part of that diencephalon that I mentioned to you earlier on. Neurons that are found in layer 5 don't send it to either structure. They send it out to the brainstem, spinal cord, as well as to another structure in the brain that we'll get to much later on, known as the striatum. So there is an organization to these, and I'll ask you to make sure that you know at least what neurons send projections out to the thalamus and the brainstem and to other cortical regions, as I've just told you here right now. And again, we can look at it in a number of ways. We'll talk about this later on. But all of these different groups of neurons, they're not also just found in a random pattern throughout the entire cortex. What we'd be able to identify is that if we were to stain them, we would actually see little striations. They're organized in these little columns. And this becomes really important, especially in vision, that all of these little columns consist of hundreds of neurons that are found in this one column. And that one column, think about it as sort of a basic processing unit in a computer. And all of those 100 neurons in that one column in one part of the cortex that you see up here are involved in helping organize how that area of cortex is going to respond to a sense of touch. So as I touch this blackboard, many of these groups of cortical columns are being activated and telling me without me looking, just by the sense of touch, many of these neurons in this area of the cortex are telling me that the blackboard is hard, that it is a rough texture, and that it's something that is cold to the touch. All of that is processed in these little mini columns. And again, I'll end with this one slide and then we'll take a break. We've been talking about this structure here at the very top. This is the cerebral cortex, often referred to as the cerebrum. It contains the gray matter, all of those cell bodies that I showed you, the 6 layers are cell bodies. They send all of their axons down through the white matter. So the brain is actually the reverse. The cell bodies are all sitting at the very outside, and they're sending their white matter projections downwards, their axons downwards through the gray. In the cerebral cortex, because it's that large head of lettuce that we stuck that brainstem or the celery stalk into, we would also find embedded within it these nuclei. Remember, nuclei are nothing more than groups of neuronal cell bodies, and we call these nuclei the basal ganglia. And these basal ganglia groups of neurons consist of things like the caudate putamen, the globus pallidus, as well as another group of neurons which are known as the amygdala. And so I'll ask you, because I think this group actually knows, the globus pallidus and the caudate putamen, what are they involved in? Or what sorts of things do you think they're involved in the brain? What do they help you do? Any ideas? So they're an output. They send information out. But what kind of information do they help you with? What types of things might they be involved in doing? Any ideas? So these are the ones that you'll see later on are involved in things like Parkinson's disease as part of the basal ganglia. The amygdala. I know some of you have taken psychology. What is the amygdala involved in? Fear. So I'm glad that at U of T you all know the fear response. That's great. You've been trained really well here. So all of these different nuclei, whether they're the amygdalar groups of neurons, the globus pallidus, or the basal ganglia, are these large groups and cell bodies that we find embedded or studied throughout the cerebral cortex. And we'll talk more about that a little bit later on. So I think now is a good time to take a break. If you can come back within the next 7 or 8 minutes, then we'll pick up and go on to the second part of today's lecture. These are the types of questions that I like asking you on the midterm. I'll leave this up here for you for a while. Think a little bit about it. You don't have all of the information to be able to answer it, but you should have most of the information to be able to answer it, and then we'll go on to the second half of today's lecture. neurons that make up the nervous system. They don't. They just know where to send the information. From a research standpoint and from a neural network standpoint, for example, if you're in research, you can't really get feedback because it's a brain structure. Only like 1 million sequences and some transcriptions to pick it up on the no, from a neuron standpoint, no difference whatsoever. Send me the email. So the reticular formation, because it's very active, we'll eventually be getting there.
So just for some clarification, you're saying that Jack's not
It's remote cortex with 6 layers which make up primarily the different cell bodies. They do some processes there for the layers, but it's right
is it a rule of thumb that white matter you would consider axons?
it's axons. It's not
Yeah.
Sorry, I totally forgot. If any of you need a hard copy of today's lecture, sorry, I just remembered I had them. I'll leave a couple of copies up here. Okay, if you don't mind, I'm going to try getting on with the second half of today's lecture. We do have a long lecture today. Thursdays will be a little bit shorter, and I have to apologize. We've got to get through all of this neuroanatomy before you can actually get into the visual system, and hopefully you'll understand the reasons why. Today I'm going to start the second half of the second part of the lecture, looking at the different ways in which we can get signals from the periphery in toward the brain, through those different pathways that I talked about, through the spinal cord, from the spinal cord up into the brain, into different areas of the cerebral cortex, whether it's the parietal lobe or the occipital lobe that we'll see on Thursday, how we can get these things into different regions of this really complex structure that is organized in this really fantastic way. So I'm going to give you an overview really briefly of the different types of sensory physiology systems that we'll be looking at. We'll be talking about specific receptors that are found on your skin, and these skin receptors, the somatosensory receptors, tell you about touch. You have other receptors that are built into your skin, also found in your body, that are related to senses of pain. They are not the same, and these different sensory systems are really important because the way they send information up to your brain is really quite different as well. We'll look at this idea of receptive fields. We won't spend too much time on it, and then we'll look at this concept known as adaptation. As you're sitting there right now, or as you're resting your arm on your tablet arm, you're probably not feeling that as well as you did when you first put your arm down. That is an example of sensory adaptation. The signals from your arm are no longer coming in there. You sort of blotted all of that out. And you need to understand that some parts of the sensory system have this capacity to adapt. And this form of rapid adaptation or slow adaptation is a big part of this somatosensory system. I'll give you a brief overview of the receptor systems that are involved in your joints. Your joints are also capable of telling you where they're located in space, even if you close your eyes. And you're able to do that without touching anything because you have built-in receptors known as these proprioceptors, things like muscle spindles, the Golgi tendon organ, as well as the joint afferents, and we'll go through those as we go along. So the general ideas that we're going to be looking at is that when we start looking at different types of sensory information, all of them have to be able to encode, whether it's a sense of smell or a sense of vision or a sense of touch. All of them have to be able to take a physical signal, whether it is the pressing up of your finger against the wall, or whether it's a photon of light that's stimulating a photoreceptor. They all have to start somewhere. And these different signals all have to work their way into the CNS, and there has to be a translation of something that is mechanical, like touching something. That is not an electrical signal, right? I'm just knocking on something. That's a mechanical signal. How did I transfer that mechanical signal of knocking on something into an electrical signal that my brain understands? And that's the process that we're going to be looking at here today. So we'll start with different things like physically distorting these receptors. So some of these receptors that are found in the somatosensory system, the two types that we're going to see are the ones that are either free nerve endings, where something binds to these nerve endings, physically compressing these nerve endings that are associated with the somatosensory system. In the proprioceptive system or the joint system, how stretching something or compressing something will activate this neuron. But all of these involve physically changing a signal into electrical action potentials that your body, your brain can understand. And we'll see these other systems like the visual system, it's the same idea. How do you get a photon of light to be converted into electrical activity that your brain can recognize? If you shine a light on someone's brain, has no effect. You won't signal a single action potential on your brain. If you have an exposed brain during neurosurgery, for example, and you were to talk to it, it wouldn't even know that you were doing anything to it. How do you convert something? You require these specialized types of receptors, and that's one of the things that we'll be looking at. And the special senses we'll see are, they have very specialized, structurally complex receptor systems, and we'll see that starting on Thursday with the visual system. So to get to where we want to go, we need to talk about Mueller's law. So these sensory systems can be divided into 5 major categories. we can either have receptors that will respond to a chemical change in the environment, chemoreceptors. It could be a change in pH. There could be a shift in oxygen. It could be a biomolecule that gets activated. They could be photoreceptors that we'll see, for example, on Thursday, where photons of light stimulate these specific receptors that cause action potentials to be generated. They could be nociceptors that we'll be talking about today, associated with pain, where you physically damage tissue. And by damaging tissue, you release chemicals. And these chemicals, which are known as noxious stimuli, then will activate receptors on that nerve. Or they could be involved in temperature regulation, like thermal receptors. Or they could be mechanoreceptors that we'll be talking about today, related to things like vibration, or acceleration or sound. All of these involve Mueller's law. And Mueller's law says that sensory perception depends on which sensory pathway is being stimulated. So in other words, if I have a specific form of energy that activates a specific pathway that is involved in the visual system, then it's going to recognize it as a visual signal, okay? So even if I don't have a photon of light causing that to be converted into an action potential, if I stimulate that neuron directly, I'll be able to fool my brain into thinking that that pathway is activated, and as a result, it will obey Mueller's law. That perception of that sensation involves that pathway being activated, and we'll be looking at how we activate that pathway later on. And again, it's shown for you sort of in this common way, in this sort of theme. You need whatever stimulus, whether it's a photon of light, whether it's the vibration of the air in terms of hearing, whether it's deformation of your skin related to touch. All of these things have to work on a specific receptor. So if it's a photon of light that's shining on my skin, no response, the receptor is wrong, we stop right there. However, if the receptor gets activated, it transduces, it changes that signal into something that the nervous system can understand, which gets relayed as action potentials through a primary sensory neuron, which is then coded and processed at the level of a secondary neuron. So it goes from one neuron to a second neuron, and then it will go through a third neuron, where eventually, in order for you to understand what's going on, to know that I'm actually touching a blackboard and not touching something else, then in order for that processing to go on, it has to be processed up in the appropriate area of the brain. So for touch, for example, I would have to activate mini columns that are involved in the parietal lobe that are related to touch. And all of that means that I'll get this sensation or this perception of what's going on in the world around me. So in other words, I have to start with this sensory transduction pathway. So I need to activate a specific sensory receptor. I need the right stimulus, and the right stimulus has to be able to activate that receptor, whatever it is. Today we'll be concentrating on the touch pathway, and then it has to be converted into a signal that can be recognized by the nervous system, and this is that process that starts at signal transduction. So somehow, by pressing up against this type of nerve ending that is encapsulated, and have you talked about this at all with Dr. Key? Like nerve endings, encapsulation of nerve endings. So this is one nerve ending, and this is one type of neuron that you see here. And this nerve ending is encapsulated. And I'll get back to this when we talk about Meissner's corpuscles and Pacinian corpuscles. But these lamella that you see here encapsulate, and it's like an onion shell that wraps around the free nerve ending. If I were to press down on these different lamella and these nerve endings, it would eventually cause this action potential or groups of voltage gated ion channels to become activated by touch, and by doing so it will start this action potential running down this axon, and then things follow along exactly like you learned in the last lectures with Dr. Key. So that's where signal transduction comes into place. We can, by changing how long action potentials fire for, by pressing longer on an object, I might stimulate more and more or have bigger action potentials. These action potentials can last for longer periods of time. All of that kind of information is encoded at the electrical level in terms of duration, in terms of how strong the stimulation is, as well as where it's being stimulated on the surface of my body. So all of these different things can get encoded. So I know like how hard I'm pressing on something, what I'm doing, and how much effort this is all requiring. So ultimately, these all rely on these different receptors that are built into your skin. And this is where we're going to start today, looking at the different things that are built into your skin. Your skin can detect things like temperature. It can detect things like pain, as well as touch sensation. And your skin has receptors that are specific for all of these different sensory modalities known as temperature, pain, as well as touch. Sometimes these receptors overlap with each other. For those of you who have an older sort of yellow HB pen or pencil, if you put it on the back very lightly on the back of your hand and you were to press down lightly without trying to break through the skin, if you were to do that, one of the things that you would find on different patches of your skin, it's not going to be the same on every patch of skin. It's not going to be the same for every individual. You'll feel the pressure, but you'll also feel something else. Can you think of what that might be? So it's a pencil. It's graphite. It's pressing down on your skin. Yes, you will feel the touch. That's one aspect. But there are two other things that you might get there, and you're pressing gently. What else might you be able to detect at the same time? Hopefully not. Hopefully no pain if you're not trying to like burrow in. Yes, please don't do that to your next door neighbor. Try not to jab them with a pencil. What else might you feel? Yes. Yes, it might be cold, it might be temperature, and that's certainly one of the things that you'll find. But one of the other things that you'll see is not every patch of skin has this overlapping modality. Sometimes you'll only feel the pressure, and sometimes you'll feel pressure plus temperature. It really depends on how these sensory receptors overlap with each other. And again, this is showing you exactly the same thing. This gets a little bit into receptive fields. A patch of skin is innervated by many, many different types of nerve endings. Nerve endings for temperature, nerve endings for touch, nerve endings for pain. And what you're stimulating when you're doing different types of things, whether you're just jabbing with your finger at the back of your hand or you're using a sharp object, it will really stimulate different groups of receptors on different patches of your skin. And this gets into this idea that when you press down, you're stimulating specific groups of neurons that have a specific receptive field. So in other words, if I press down on this patch of skin that is innervated by this red group of nerve endings that are attached to this one red neuron, I will only be stimulating this one red neuron. At the same time, if this other blue neuron right beside it is also being activated, I might activate this blue neuron as well because its receptive field overlaps. However, this other neuron that is outside of the receptive field that's found by this red neuron and this blue neuron, its receptive field is only made up of these nerve endings, and you wouldn't activate that one. And so you would only activate 2 out of the 3 receptive fields here. You wouldn't activate all 3. And so you will have like really good discrimination of the difference between pressing down really hard and activating all 3, or activating just one group versus activating 2 of these. So this is one of the ways in which we can localize, using these receptive fields, different types of somatosensory touch on different patches of skin. All of these different types of modalities, by the way, whether it's touch, temperature, or pain, all of them send their projections up to the thalamus. Do you guys remember where the thalamus is in the brain? The diencephalon. Perfect. Okay, so the diencephalon, one of the areas that we talked about was the thalamus. This is the relay center of the brain. All of these information is being sent up through the spinal cord toward the thalamus. Okay, and the sensory signals that come from the skin have the property of decussation. Have you guys talked about decussation at all? Do you guys know what decussation is? Yes, no, maybe? So some of the signals, as you know, go from one side and stay on one side of the body. Some of them cross over. And when I say that the information has decussation, it crosses over from the left to the right, or from the right to the left. It crosses over, in other words, and this is one of the things to keep in mind. So if we were to take a look, and this is an important slide for you to go back and review. If we were to take a look at this sort of unique ganglion cell, and we talked about these ganglion cell, and we talked about the dorsal root ganglia. When we get a whole bunch of these together, and we look at the ganglion cell bodies, it's the dorsal root ganglia. So imagine that one part of this dorsal root ganglion cell sends its axons out to your skin. It has these free nerve endings embedded in your skin, and the other part sits near your spinal cord, and then it will eventually send its signals through what part of the spinal cord into the actual spinal cord neuron. So it's sensory information. Does it go into the ventral region or through the dorsal? So through the dorsal region, it will activate this particular spinal cord. So this is what's known as a pseudo-unipolar ganglion cell, just by the type of shape that it has. And one of the things that you can do is you can, by pressing down on the skin or electrically stimulating it, you can generate what's known as a generator potential. This area of the neuron will start to become electrically active. If it reaches threshold, by the time it reaches this first node of Ranvier, it will actually just start generating self-propagating action potentials, which will then spread all the way back down, go past the cell body, and will eventually cause the release of neurotransmitters onto this spinal cord neuron, just like you learned in the last series of classes. One of the things that makes this respond to touch is that there are cation channels that are embedded here at this receptor end. Okay, so we have cation channels down here. When you deform these things, cations flow, cation channels open up. So go back to what you learned with Dr. Key. Cation channels. If I now open up these cation channels, not by changing the membrane potential, but by physically pressing down and physically deforming the channel so that they open, same idea, cations flow into the cell down their concentration gradient. What will happen to this neuron? It will start to depolarize. That's 100% correct. It will start to depolarize because of these cation channels. It will, if it reaches threshold, it will generate these action potentials, and it will allow for this propagation to occur in the same manner using those voltage-gated sodium channels. Again, if we were really lucky, it will also stimulate the spinal cord as well. Do you have any questions about this before I go on? Because this is really the way in which all of the different sensory systems that we'll be talking about work in this manner. Okay, so all of these somatosensory receptors, as I told you, we have 5 different types. Somatosensation, when you're touching something and you're converting it to an electrical signal, all of them are these mechanoreceptors, okay? It's not a chemical receptor, it's a mechanoreceptor, and there are 2 broad categories of these mechanoreceptors that are telling you about a sense of touch, pressure, etc. These are cutaneous receptors that are found directly on the skin, detecting changes in pressure and stress, and these proprioceptors that detect changes in muscles, joints, as well as tendons. Okay? So please make sure that you're able to keep these 2 separate. We'll be concentrating on these cutaneous receptors for the most part, and we'll be talking about these proprioceptors at the very end of this particular section. Okay, so this is what the overview of what the different sensory signal receptors are in your skin. So embedded on the glabrous or the smooth, hairless part of your skin, so on your palms, on your fingertips, for example, we have these peripheral receptor systems. They're all embedded at the level of either the epidermis, just under the epidermal layer, all the way down to the dermal layers, the deeper ones. And I'm going to ask you to make sure that you know, for example, on this slide, the deepest one that you would find in the dermal layers, so below the surface of your skin, before you get to the layer of fat, the subcutaneous layer, you will find this specialized receptor known as the Pacinian corpuscle that you see encapsulated with this onion-like shape that is known as the lamella. The next layer up, we'll find these Ruffini corpuscle endings, and then we will find these other Meissner corpuscles at the level just under the epidermal lining. All of these different types of receptors that we find here tell you about different types of things that are going on the surface of your skin. Okay. So one of the things that's really important about this is not only can we identify things related to the cutaneous receptors, we can also identify things that are related to the size of the axons that are accompanying these different types of receptors. So one is known as the A alpha slash beta. These are large diameter, these are myelinated axons. So when they're found with encapsulated receptors, the Pacinian corpuscle, for example, is an encapsulated receptor. The Meissner corpuscle is an encapsulated receptor. These signal at about 50 meters per second. Is that fast? Yes, pretty fast. Okay, good. So in other words, they signal very quickly toward the spinal cord. The A delta, smaller diameter, myelinated. These are often associated not just with mechanoreceptors like the A alpha beta, but these are also associated with thermal receptors. They're the axons that are also found with the free nerve endings that allow you to tell if something is cold or hot. These are also the types of axons that are found with the things that tell you if something is painful or not. And these signal at about 10 meters per second. And the receptor type that they're associated with is the free or naked nerve ending that is just under the epidermal lining of your skin. The last type of cutaneous receptor, the axons are much smaller. They're the smallest diameter. They're also unmyelinated. And as you guess, smaller diameters, no myelination. These are the slowest of all of the different systems. How fast is 50 meters per second, by the way? 5 times faster than? Oh, 5 times faster than Usain Bolt. Okay, good. I guess so, sure. Um, how about in real world terms? I've never seen Usain Bolt actually run, except on TV. What does that really mean? 50 meters per second. How about miles per hour, kilometers per hour? I drive fast. I drive like 120 kilometers an hour. Is that faster than this? Probably not. So this is how fast that signal gets from where it starts to where it gets into your spinal cord. So if you ever have the opportunity to watch IndyCar racing and you sit there and you're watching like one patch of the roadway and the car goes by, that's how fast your nervous system has just signaled. Versus this, the 1 meter per second is really, this is how fast that signal has gotten into your spinal cord. I am walking right now 1 meter per second. Your spinal cord is not seeing that information very quickly. That pain signal, for example, that is being transmitted, as soon as I stub my toe, that signal goes at 1 meter per second. It's very, very slow. If any of you have ever done that in the dark where you stubbed your toe and you felt the pain, sometimes you know that, oh, this is going to hurt really bad before it actually starts hurting. And that's why that signal is so slow before it actually starts. C unmyelinated small diameter fibers transmit information to your spinal cord and into your brain extremely slowly. And so that actually is something that we can make use of a little bit later on. So this is one of those somatosensory receptor systems, the Pacinian corpuscle. I'm going to go over this section down here very, very briefly. I'm not going to spend a lot of time on it this year, but I will tell you a little bit more about this Pacinian corpuscle that's found in the deeper layer, in the dermal layer. This is the largest cutaneous receptor that we have. If you have these deeper in your skin, for example, they use those large myelinated axons. They transmit information incredibly fast, like those IndyCar going down the speedway at 250 kilometers an hour, roughly around that speed. They have a very, very large receptive field. One of these Pacinian corpuscles is enough to innervate like a huge region in the palm of your hand. You don't need very many of these to let you know that something is signaling to your dermal layers. They're located deeper in those dermal layers, as I showed you. Typically, we have them on the palms of your hands, on the soles of your feet that allow you to signal that something is happening. Pressure is being applied to your hand. These are what are known as rapidly adapting. And you guys have covered that with Dr. Key, is that correct? Rapid adaptation, fast adaptation. Okay, we'll see if you remember all of that in a few moments. And these respond best to a skin vibration of 200 hertz. 200 cycles per second. Something is vibrating at 200 cycles per second. That is what stimulates these receptors the best. Think a little bit about that. Where do you encounter things that vibrate? I actually don't want to know. At 200 cycles per second. That means these are very, very sensitive, by the way, and they have a threshold. Even though they have a really large receptive field, you can tell with a 0.5 micron indentation. In other words, if you press a leaf against the palm of your hand, it's that receptor that's actually being able to detect it, but it rapidly adapts. It turns itself off really quickly. So this again is a form of rapid adaptation. This would be, if we were measuring from a Pacinian corpuscle, this is what we would most likely see. If we were to take a patch of skin that we know has in it a Pacinian corpuscle, and we started to apply pressure to the Pacinian corpuscle that supplies this patch of skin, and we increase the pressure, we keep that pressure on during the square wave pulse, and then we let it go. We take the pressure off, and it goes back down to where it was before. This is what we would see if we were able to measure the Pacinian corpuscle. We would see that it rapidly adapts. In other words, when we turn on the signal, when we start pressing down, it starts firing action potentials like crazy. But while we have that pressure on, as long as it's on, there is no more action potentials being generated. I take that signal off, and then it starts firing again very briefly. Is that okay for everyone? So this rapidly adapts. Does it really give us much useful information? It's really sensitive. It would be really useful if we had something that had a 200 hertz cycle that was stimulating your skin. But we really seldom see things that have this type of stimulation pattern, right? Or do we? Can you think of something in the real world that would stimulate the Pacinian corpuscles that might lead to something that looks a lot like this? Any ideas? So why do we have these things? So yes, they'll tell me that I'm pressing my hand, but then as soon as I start pressing, this thing should theoretically turn itself off. Why bother having this Pacinian corpuscle? Any ideas? So look at your hand. You don't have to look at your hand, but you know, if you were to look at your hand in detail later, you'd see that there are ridges on your hand. It's not smooth, right? Even though it's smooth skin, there's no hair on the palms of your hands, hopefully. If you were to look at it, it's smooth, except you have ridges on it. Every time you move something against that ridge, you're actually creating a vibration that will activate your Pacinian corpuscles. So imagine you have something that's actually slipping out of your hand, where it's activating those receptive ridges. Your Pacinian corpuscle is able to detect that. In other words, when something is slipping out of your grasp, these Pacinian corpuscles are the things that get activated. And yes, they have these massive receptive fields. One Pacinian corpuscle is detecting any force applied in this broad area versus a Meissner corpuscle. In order for me to stimulate a Meissner corpuscle, I would need to be able to have a very small stimulus, not this wide swath, but this narrow, small stimulus to be able to stimulate a very different looking somatosensory receptor known as the Meissner corpuscle. So Pacinian has this really large receptive field. No matter where I press on this area of the hand, it's still the same Pacinian corpuscle. If I stimulate here on this area of the fingertip instead of the palm of your hand, it's that one Meissner corpuscle, and as soon as I move off of it, it's a different receptive field. It's a different Meissner corpuscle that will be activated. And again, this shows you that the receptive fields are really important. So much, much smaller for Meissner's corpuscles, and very similar for Merkel's discs. Again, this gets into this idea that on different patches of your body, you have different receptive fields. You don't have to memorize these values. The bigger the bars are here up at the top, the larger the receptive fields are, so the fewer receptors you actually have there. And the smaller, the lower these bars are, the more receptive fields, the more receptors you have actually in that area of the body. So you have lots and lots of receptors in your hands, for example, and you probably know that. You have lots of densely packed somatosensory receptors in your hands, and you have very few on your shin or on your knee, which means that you have a less sensitivity on what's known as a Lyman 2-point discrimination. So what they do is you simply get blindfolded. They put 2 probes down and they ask you, can you feel 2 probes or 1 probe? And you might think, wow, that's a silly experiment. Of course, if you put 2 probes on my knee or on my leg, I'll be able to detect that. Not if you're blindfolded. So if you don't know, you'll still feel like you're pressing like 1 probe down on you. Except when you get to your hands, then you'll be able to know like right away that because the density of the receptive fields is so high that this 2-point Lyman becomes really quite sensitive. And the worst areas, again, sort of at the legs, back, as well as your arms, where you don't have as many receptive fields. So another really important cutaneous receptor are these Merkel discs. Unlike the Pacinian, which is rapidly adapting, this is another sort of cutaneous receptor, somatosensory receptor, and these Merkel discs are found on your skin, just under the epidermal layer, and these are the slowly adapting types. So they stay on much longer after you start pressing and stimulating a patch of skin. Again, they are also very, very sensitive. Not quite as sensitive as Pacinian corpuscles, but 20 microns. Again, you only have to indent your skin less than the width of a piece of paper in order to activate these Merkel discs. It doesn't require a lot of pressure, in other words, to activate them. These again, if you start stimulating the axon, you take an electrode and you directly find an axon that is coupled with a Merkel disc. It's a lot like feeling a leaf or feeling a puff of air on the back of your hand or on your fingertips. And this is what it feels like when you activate the axons here. There's much greater innervation on the fingertips. It's very much like the Meissner corpuscle that I showed you. So you get lots of these along with the Meissners on your fingertips. Like these Merkel Meissners are also there. This provides you with greater spatial acuity overall. We have a lot more of these per square patch of skin than the Pacinian corpuscles, and they also have a smaller receptive field. So women do have a better sense of touch overall. So women have smaller fingertips, and they have the same density of these Merkel discs, and so they have the ability to have a finer sense of touch, and they have a better sense in terms of adapting to surface material, and they're able to identify things a lot better because of the sensitivity of their Merkel discs. So that's one type, somatosensory. Again, we've concentrated on your patch of skin. You have somatosensory that is involved in touch. We have pain receptors that we haven't talked about. And the other thing that your skin can detect is temperature changes. So when we look at the same patch of skin, as I told you before, sometimes they overlap. That same patch of skin can also contain thermoreceptors. So it's not just one type for this patch of skin. It could have like many other types of receptors as well. These are also slowly adapting, just like the Merkel discs that I told you about, slowly adapting. These are not encapsulated. And by the way, I have to tell you, these Merkel discs, as the name suggests, are encapsulated. They have a disc-like lamella around it, not this onion-like structure, but one large disc, so they are encapsulated endings. But thermoreceptors, generally speaking, like pain receptors, are free nerve endings. There's nothing around the nerve terminal at all. Not a disc, not lamella, not this corpuscle. It's just a free nerve ending. And again, we can identify these into different classes. So there are patches of your skin that respond better to warm sensation. These will use, these are the group C type of thermoreceptors. And just like we learned previously, these group C are the small unmyelinated. They transmit information at very slow temperature, slow conduction velocities of about 1 meter per second, versus the group A delta, which are responding best to cool temperatures. These again fire at about 10 meters per second, and their axons are small, but they're also myelinated. And they respond to different temperature sensations. So your patch of skin might have warm receptors that respond best at temperatures between 25 and 45, and you might have another group that respond best at 10 to 28 degrees. And it might not sound like it's all that cold, but in fact, outside of these ranges. So if you've ever gone to Tim Hortons or you've gone to Starbucks, that cup of coffee is not boiling hot. That cup of coffee that you're picking up that you feel really uncomfortable with just has to be above 45 degrees for you to feel pain. So outside of that range, you're actually activating pain receptors. Below, for example, 10 degrees Celsius, you're actually activating polymodal receptors that respond in a painful way to cold stimuli. So again, these are slowly adapting. They have a phasic component. And when I say phasic component, do you understand what that means? Yes? No? What does phasic mean? And it's not a term from Star Trek. Okay, so I'll show you that on the next slide. Hopefully it's a review, and maybe I'm just using a different term than what Dr. Key told you earlier. So these are free nerve endings. They're slowly adapting. Again, they fire at different temperatures. And one of the ways in which they activate and cause electrical activity to occur is because on these free nerve endings, embedded within them, we have what are known as these transient receptor potential, or TRP cation channels. These are non-selective. When they open up, with the proper stimulation, these TRP channels that are found on these nerve endings will allow both sodium as well as calcium to influx, then causing that action potential to propagate down and stimulate into the spinal cord. So this is what I mean by phasic as well as tonic activation. So this phasic component that I mentioned to you before, I'm going to show you this on this particular slide. So here's a patch of skin, and on this skin, one of the things that you can do is you can add a temperature probe, and this is how they do this. They will add a temperature probe that is either going to be hot, somewhere between 40 to 45 degrees, versus being somewhat on the cooler side, for example, below 21 degrees. So if I took this one patch of skin and I applied this temperature probe, your skin is sitting there at 34 degrees Celsius. So it's because it's being exposed to the air. Then you apply this 40-degree probe to the back of your skin that you're recording from. And as you do so, and you leave it on for this period of time, at the same time, you are recording the activity in the neuron that's associated with this patch of skin. And one of the things that you would see at 34 degrees, it would fire an action potential, fire an action potential, fire an action potential. And then as soon as you put the hot probe on, 40 degrees being, again, something that stimulates the warm receptor, then you would see it firing. And there's a burst of phasic activity. It's phasic. It becomes activated for short periods of time, and then it gets into this sort of rhythmic activity. It has adapted to this sort of probe being on it, and then when you take it off, it goes back to its baseline recording. The cold receptor, if you were to record from the same patch of skin that's being stimulated, but instead of measuring from an axon that is responding to warm, you are measuring from an axon that responds to cold. During this time that you've applied this particular type of stimulus, remember, it's the right stimulus to the right receptor that causes the action potential to fire. Nothing happens. So it's not responsive at all until you take the hot probe off, and then the cold receptor will have a phasic burst of firing, and then it goes back to complete silence. It's not activated at 34 degrees. If we do the exact opposites, the same patch of skin, now we go in, and that same patch of skin, we add a cold probe where it drops the temperature down to 21 degrees. The warm receptor that previously gave us this response now shows exactly the opposite. During the time that this cold response is on, it's the wrong signal. The receptor cannot respond in the appropriate manner. The axon is not being activated until we remove this. It goes back up to the normal temperature, then it fires, and then it starts its normal rhythmic activity at room temperature. The cold does something completely opposite. It fires normally at 27 degrees below the normal room temperature, your normal body temperature. And then as it gets stimulated more, it has this phasic response, and then it has this static response as long as the probe is on. And this is why you respond to different types of stimuli on the back of your skin. Sometimes it's a phasic response, sometimes it's a static response, but all of your skin is innervated by these different types of receptors. And again, this is just to give you an idea. On a patch of skin, for example, you might have these cold polymodal nociceptors. You might have cool thermoreceptors, which I just showed you. I haven't spoken to you about these cold polymodal yet. The cool thermoreceptors that respond best somewhere between 10 and about 27 degrees, and the warm thermoreceptors, and then these hot nociceptors beyond sort of the 42 to 45 degrees. Then you activate a different class of receptors altogether. And so I'd like you to make sure that you are aware there's actually very little overlap between these different types of receptors. You don't really get a lot of overlap between the cool and the warm receptors. They don't respond to the same stimuli on the same patch of skin. And you also don't have a lot of overlap between the warm thermal receptors saying that, yeah, it's nice and warm, versus another group of free nerve endings that say, wow, this is actually really painful, and you should be looking at responding to this. So this gets into the last class of the somatosensory receptors, known as the nociceptors. These are the pain receptors that are found embedded throughout your skin. They again are classified according to their the size as well as the myelination of the axons that are associated with the receptors, all free nerve endings. This is the group A delta, the cool thermoreceptors. They are also associated with high threshold mechanoreceptors, and these are involved in prickling types of pain. The group C, the unmyelinated ones, respond best to warm, and they're also known as the polymodal nociceptors. And these are the ones that respond to tissue injury. When you physically have damaged your skin, you've bumped your toe, you have cut your finger, all of these types of pain that damage your skin, for example, signal through these polymodal nociceptors that lead to tissue injury, that give you that sense of burning pain. Whether it's a stub toe, whether it's a cut finger, whatever it is, that burning pain that you feel is being signaled to your brain very slowly, again, 1 meter per second through those group C axons. And in many cases, it's because locally in that area that you've damaged tissue, you've created a brief burst of acidity that's being recognized by these free nerve endings. There's this last type that we don't really know a lot about, which are known as the sleeping nociceptors. These have no response whatsoever to any mechanical stimulation. You can't poke your skin in any way and activate these receptors, but they will respond to some chemicals. And we think these sleeping nociceptors are the ones that are actually there to respond to sensations that give you things like itch. When you feel itchy, for example, we think you're activating these sleeping nociceptors. Again, these are all free nerve endings. Every last one of these are free nerve endings that are associated with different sized axons. So what stimulates them? I told you that it requires tissue damage. So if you have tissue damage locally, and on these free nerve endings, you have the appropriate receptors. You have receptors like ENaC or DEG. Or you have TRPV1, any of these receptors that are found on the free nerve endings that are responding to pain. When you damage your tissue and you release things like hydrogen, for example, acidity locally, or you release substance P, all of these will interact with these receptors and then cause them to become activated. It will cause the associated cation channels to open up. So how does it do that? People didn't know for a very, very long period of time. All they knew was that somehow these different receptor systems, whether it's ENaC, I didn't show you DEG here, but it's very similar to ENaC, and TRPV1, they respond to things where you physically damage your tissue. So it could be like burning your hand, or it could be like cutting your finger, stubbing your toe. All of these things get released and activate these receptors. It could be activating them through capsaicin. If you eat hot food, you are literally burning your mouth. And that burning feeling that many of you enjoy having is actually stimulating these pain receptors that are found in your mouth, for example. We don't know a lot about this last class. This last class, on some free nerve endings, have another protein channel which is known as TRPM8. We don't know a lot about TRPM8. We don't know how it gets turned on. We don't know, it doesn't activate by substance P, capsaicin. It doesn't activate by acidity. We don't know what really turns it on. But we know that if you have responses to cold, you need TRPM8. And we don't really understand how that all works. We do know that if you activate TRPV1, and it's very similar for ENaC and DEG, but I'm just showing you TRPV1. When you have tissue damage, one of the things that things like substance P, capsaicin, prostaglandins, and other things will do will be to activate a bradykinin receptor. And this bradykinin receptor is a GPCR, which will then activate and turn on PKC epsilon through phospholipase C activation. And phospholipase C will open up this TRPV1 channel. So when it's open, then sodium and calcium flows in through those free nerve endings. And when those free nerve endings have sodium and calcium flowing through, then they start being activated. You get that graded potential. And then if it hits that initial segment and activates a voltage-dependent sodium channel, you'll get action potential propagation. So ATP, bradykinin, hydrogen, substance P, all of these things will activate this TRPV1 pathway. Okay, so let me just see how I'm doing for time. I'm going to go through 2 more slides and then we'll sort of take our second break. So again, I'm not going to spend a lot of time on this. It's really just to give you an idea that these different types of receptor systems, whether they're the A delta or C fibers, respond to a variety of different noxious stimuli, either below freezing or these polymodal C fibers, which are the majority of the C fiber class. And the ones that I've highlighted here for you in the different colors are the main facts to take away. So the fact that you have A delta and C fiber nociceptors, and that the polymodal C fibers are the majority of the C type of fibers, respond to different types of stimuli. So these up here at the top respond best to mechanical stimuli or to very, very cold temperatures. And again, if you're thinking without looking back, on those free nerve endings, what type of receptor channel should we have on this system that's responding to cold? So do I have TRPV1? So it's probably that TRPM8 that is found on these free nerve endings that are associated with this, this cold sort of sensation. And these polymodal C fibers, again, respond to both cold and noxious heat. And these are very unique, and these tend to have all sorts of different receptor proteins associated with them. So the last type, these are not found on your skin. This is part of that original slide that I went through. The proprioceptors allow me to tell where my arm is in space with my eyes closed. It allows me to tell you how I'm standing with my eyes closed, and it gives me a sense to where my joints are located in space. So in other words, I need a receptor system built into the muscle system that lets me stand up, that lets me keep my balance. And these receptors are located in these different muscle tissues, tendons, as well as joints. And we'll talk about the muscle spindle organ in a moment. We'll talk about the Golgi tendon organ in a moment. These are very similar to what we talked about with A delta and C fibers. These are group A and group 1B types of afferents. And I lied to you. I'll go through one more slide after this just to give you an idea of what A alpha, 1A and 1B afferents are. And then the joint receptors that we won't spend a lot of time on as of today. So this group 1 is very much like the A alpha. They're very large diameter, myelinated, not with the skin, but with the joints. They're found in the joints. And if you were to take apart someone's muscle, look at specific receptors that are embedded in the muscle, you'd find an axon. That axon would be large diameter, it would be myelinated, and it would be responding to stretch on the muscle. And this is actually fast. It's 75 meters per second. It's very similar for the 1B, and these are the fastest types of nervous system transduction that we have in the body. So when you're reading different textbooks and they're saying that it only takes like a brief period of time for the signal to get from the periphery to your brain. This is what they're actually talking about. It's this type of axon that they're mentioning. Group 2, again, a little bit smaller, A beta type, and A delta, small diameter myelinated, and then the small diameter unmyelinated look a lot like the C type. We're just not talking about skin, we're talking about the proprioceptors and the receptors that are embedded into either tendons, joints, or into the muscle directly. Okay, and we'll see what that looks like after the break. So we'll take our break now, and we'll come back and talk about proprioceptors.
Sir, are you going to post these types of questions on the portal? Okay.
Sorry, just out of curiosity before I get going, how many of you log into your PSL Facebook group regularly? Okay. I will be sending the links to the recordings and everything else, including the voice recordings, to someone who will post them for me. If you have any other questions related to the material, please let me know. Drop by office hours. I know there's a lot of material and there's more to come. And it gets a little bit easier with the visual system, but since your midterm is next Tuesday, you don't really have a lot of the time of luxury of studying. So I know that you're in for a little bit of a cram session in the next little while. Let me know if I can help in any way, including having a midterm review on Monday or even Tuesday afternoon. Okay, so this is sort of where I ended off. Actually, at this point in time, is anyone feeling a little bit thirsty? A little bit thirsty, okay. Actually, I need someone who has short sleeve shirts on. Sorry. Yes, I'll take you, Christian, thanks. Okay, so not right now. Oh, yes, okay, thanks, sorry. So we're going to be looking at a response. It's actually physiologically really relevant. We're going to be looking at these muscle spindles. These are part of the proprioceptors. Your muscles that make up the way in which you move your joints, or the muscles that are surrounding the different parts of your limbs that are connected by joints, no one likes having their muscles pulled. So if you pull someone's arm, the natural response is to actually pull away. Right? So if someone comes up behind you and grabs your arm, your first response is you want to pull your arm away. And it's because you don't want your muscles being stretched. And part of that response involves these muscle spindles. And these muscle spindles, if this is the belly of the muscle, like in your bicep, and if you're pulling on someone's bicep, one of the things that happens is that it stimulates receptors that are embedded within the muscles directly. And these specialized sensory organs or sensory receptors are known as muscle spindles. And we have this intrafusal fiber, which consists of this chain down here. And surrounding that, we have this nerve terminal. And this nerve terminal winds its way around and will tell how much tension is on your muscle. So if you have more tension on the muscle, it knows that it has more tension. If you don't have as much tension on the muscle, then it will also know that as well. And these intrafusal muscle spindles are your receptors found within different joints, especially within your muscles. These are the ones that have those really large axons. So the axons that they're connected to that signal toward the spinal cord are those really large diameter, myelinated, super fast conducting, 75 meters per second type of axons that you see here. So they respond to any change in your muscle. These are very, again, large diameter, fastest conducting, very slowly adapting. In other words, as long as your arm is being pulled, your response is to actually pull back away from it. It continually stays on. It doesn't adapt in a rapid fashion like the Pacinian corpuscles that we saw earlier on. So if we stretch these intrafusal fibers, and how do we do that? When does that happen physiologically? Why is this relevant for you? Then it excites these muscle spindle afferents. And now I would like, Christian, if you're okay with it, if you don't mind volunteering. So how does that work physiologically? When do you actually, I'm not going to hold your hand or pull your arm, so you don't have to worry. So one of the things that happens is, how does this system work? How do you actually pull on a muscle spindle? So this is how you pull on a muscle spindle. So if you can put out your hand, and this is a relatively heavy glass bottle, so please try not to drop it. So one of the things, if you're looking at Christian, so he was able to adapt because this is a heavy weight. It's pulled down on his muscle. Why didn't he drop it? And why didn't he over-contract it? It's because this muscle system has allowed him to contract his muscle. Thank you. That's all I need you for. Thanks. And enjoy your lemonade. Thanks. So as soon as that weight has been applied to Christian's arm, his outstretched arm, he didn't know how much weight was going to be dropped onto his hand. That actually stretched his muscle out. and that pull on his muscle, that weight that you have, and I could have kept putting more and more bottles of lemonade into his hand, it would have stretched out that even more. And as long as it didn't overexert his muscle, he'd be able to maintain it in space. It had pulled this applied stretch here, meaning a weight was put on his hand. And one of the things that happened, as soon as that weight was pulled on his put on his hand, it stretched out his muscle. That sent a signal back that said that a stretch response was occurring. It signaled really, really rapidly into his spinal cord, went up to his brain equally quickly, and it said a stretch response was occurring. This did not adapt. It did not disappear. We still see that action potentials fire. But one of the things that happened, though, was that Christian actually contracted his muscle. Again, I don't know how big his muscles are, and I don't know if you could see that at the back, but one of the things that happened was when it stretched, his muscle contracted back. And one of the things that happened was this muscle, as it contracted, what do you think happened to that spindle? Is it still being pulled taut? Is it still tight? No, the muscle has contracted. This is sitting inside the muscle. The muscle has now become shorter. It's contracting. This fiber that is normally supposed to respond to any stretch has now sort of not been tight. It's not sensitized anymore. And so it stops firing at this point, which is what you see here. That muscle spindle, when we're recording from it, has stopped firing. It's no longer activated. But that's a bad thing, right? So if this does not get sensitized again, if it's really just stopped at this point, as soon as the muscle contracts, the next time I add something to Christian's hand, in this instance, he won't know if his muscle is being stretched again. Again, another weight being put on it. So one of the things that his body has done, again, through the nervous system, is that it has caused a resensitization through a gamma motor neuron fiber to actually pull that chain, this chain here. It has actually caused it to be pulled tight. It's actually also contracted as well. So his muscle has contracted, and so has that intrafusal fiber. The intrafusal fiber itself that's sitting in there through a gamma motor neuron has also been resensitized, and that occurs within the order of microseconds. So if I had put one weight on Christian, stretched out his muscle, it would have responded. And within a few microseconds, if I added another weight, he'd still be able to respond because he's reset this whole system, going back into that resensitized state. So this is why we talk about the nervous system and why we have to worry about having these different types of myelinated, large axon, rapidly conducting. You can imagine every time you're standing there on the subway, and the subway is causing you to shift your weight back and forth. If you're not able to resensitize, you'd be falling over flat. Your muscles and your legs have the same sort of receptor systems that you have up in your biceps. And with the shift in weight, if you're not able to adapt and resensitize the intrafusal fibers in your muscles, you wouldn't know that you have to be standing upright the next time a heavy load yourself shifts in terms of weight on the subway. Again, we won't worry too much about the different types right now, other than these are really rapid in terms of signals into the spinal cord and up into the brain. And again, these respond very, very quickly in terms of the velocity as well as to the degree of stretch. The more I stretch it, the more strongly it contracts back. You don't actually have to worry about it. It's a spinal cord reflex that we'll talk about down the road. And again, we have other ways to determine, and if you're thinking about this, how did Christian know not to over-contract his muscle, right? So why didn't he drive that bottle of lemonade into his eye? Like, why didn't he just like over-contract and like drive it in there? And part of that involves another system that we'll talk about later, and this is the bottom half that I'm showing you now. There is a compensation mechanism done at the level both of the spinal cord as well as in higher levels in your brain. So for the time being, we'll just worry about understanding the muscle spindle, the encapsulated muscle spindle, the fact that it fires through that type 1A afferent, which is a large diameter myelinated axon, and that allows you to adjust to muscle stretch, which means that you contract, and in order to resensitize things, you need that gamma motor neuron to cause the intrafusal fiber to also contract and resensitize. So that's in your muscle. You also have other types of specific receptor systems in your joints. And these are, again, are part of the proprioceptors that allow you to know where your joints are or your muscles are in space. And these are specialized types. These attach to capsules that envelop a joint. So you'd find it at the joint, for example, in your arm or in your knee, in your shoulder. And these are smaller, group 2 as well as group 3, medium and smaller diameter, all myelinated axons. These are not sort of the encapsulated types of intrafusal fibers that I just mentioned that are found in the muscles. Here we actually have Pacinian form. In other words, we have those rings of lamellae, and these are known as Golgi-Mazzoni corpuscles that are found embedded directly within your joints, and they can tell if your joints are being stretched or compressed orthogonally at 90-degree angles to how the joint normally should be. And again, we have other types of endings, Ruffini endings, that respond to when your capsule, the joint capsule, is being put under heavier stress. So if you're bending or pulling the muscle, you're also stimulating these Ruffini endings as well. So we have a number of systems in the muscle, in the joint, different types that respond to different types of stimuli that allow you to know what's going on with your arms and limbs. So we talked about things on your skin, temperature, touch, as well as pain. And then we moved into what happens with your joints, and we talked about the different systems that are involved in your joints. And so we talked about a lot of things today. Make sure that you spend some time over the weekend to review it. If you have any questions before the weekend, please let me know. I'll be happy to go through it with you. And now we're going to talk about something related to all the things that we've talked about. So we've talked about what happens out here. We talked about how the spinal cord is organized, but we haven't really talked about how information gets from out here into the spinal cord, other than looking at receptor endings and how action potentials might be generated. There are specific pathways in your spinal cord. Remember, how many funiculi did I say that you had in your spinal cord? 6. Perfect. So now we're going to look at some of those different pathways that are found in your spinal cord. And do they all convey the same information, or do different regions convey different types of information? By the way, you don't need to worry about things related to Neuro 101. It's an older textbook. If you need anything specifically about what to study, please come and see me, or please email me if you can't come down to campus every single day. I'll be happy to go through things with you electronically. But we're going to be talking about how we get information from the periphery, from those receptors into the spinal cord, and then from the spinal cord up into your brain. One of the things that we'll look at is this specific pathway for touch. And this touch pathway, the somatosensory touch pathway that goes from your receptor, whether it's Pacinian or whether it is a Ruffini ending or whether it's a Merkel disc, etc., how it goes in your spinal cord on the dorsal column and then gets into the brainstem at the level of the medial lemniscus, and how it then gets into your brain, into different association areas like cortical area 3B, for example. And then we'll talk about the pain pathways like the spinal thalamic tract and the spinal reticulothalamic tract. And again, a lot of it is going to be neuroanatomy, so apologize in advance. And we'll talk about how these different pathways can actually be physiologically relevant for you. So this is all the different pathways that we'll be talking about in a little bit more detail, so it's useful to come back to this slide if you get confused a little bit later on. When we talk about touch, it's this dorsal column medial lemniscus pathway. Anything dealing with touch, Pacinian corpuscle, Merkel disc, anything that is a cutaneous receptor on your skin, it's really about these dorsal columns. These are all mechanoreceptors. These are relatively fast pathways. These have these small receptive fields, except for the Pacinian corpuscle, which has the large receptive field. If you're talking about conveying information related to pain or related to temperature, it does not send information up to your brain in the spinal cord in this the dorsal column medial lemniscus pathway. It sends information up through the spinal thalamic pathway. So it's a totally separate pathway that leads up to your brain into a different area in your brain that allows you to process information related to pain or temperature. Okay, so these are different. This is mechanoreceptive, somatosensory, this is pain, different area of the spinal tract, different area in the brain. So make sure that you are comfortable with both of these different tracts. And the spinal reticulothalamic is the one that is related to pain. It's the slowest. It will actually have mixed modalities, including pain, temperature, etc. And the last thing that I'll mention as we go through this today, when we get to this particular section, in the spinal thalamic tract, we'll see all sorts of very specific types of neurons in the intermediate zone. And one of those is known as the wide dynamic range neuron. I'd like you to make sure that you are familiar with the fact that it's very common to find this type of neuron in the spinal thalamic tract. And I mentioned that here, and I'll mention that as we go along as well. This is a really busy slide. You don't need to memorize all of the details on this slide. For example, I don't need you to know the difference between the gracilis versus the cuneatus. These are ones that convey information from the arm versus the one that conveys information from the leg. It's really just to give you a roadmap of how this thing all works together. And again, the more important information is here at the top. So we have these Meissner corpuscles, we have these Merkel discs, we have these Pacinian corpuscles. We haven't really talked about Ruffini endings or Ruffini corpuscles. All of these, though, use those A alpha, A beta type of axons, large diameter myelinated, and they send their information related to fine touch and vibration directly into the spinal cord. So what happens is, when I say ipsilateral, what does that mean to you? So it's ipsilateral here on this side. What does that mean? Any idea? Same side, that's right. So if I'm touching things using my Merkel discs, Ruffini corpuscles, etc., all of that on my right side, that information stays on the right side of my body as it gets into the spinal cord. So it stays on the same side as where I'm getting this physical information or this touch. And it stays on the right side of my spinal cord. All that information is being sent up through this dorsal column medial lemniscus pathway in the spinal cord that I'm going to show you in a moment, in the same side. It's on the right side all the way through until it gets into the medulla. At the level of the medulla, once it gets past this dorsal column pathway, once it gets into the medulla, then it decussates. It crosses over to the other side. That information that's come up along this right side of my spinal cord all the way up to the medulla now has crossed over to the left side of my medulla, and all that information from the right side has now gone on to the left side of my thalamus and will go into the left side of my cortex. It crosses over. And this is where a lot of these crossing over events happen. So again, this gets into that primary, secondary, tertiary level of neuronal responses. Here's that primary neuron, that secondary neuron, and then that final group of neurons that gets up into the cortex. And we'll talk about this dorsal column in the spinal cord medial lemniscus pathway that goes up into your brain in a second. Do you have anything that you want to clarify, what you need to know, or anything else on this slide? It's a really busy slide, but in many ways it's just an overview slide. So other than what I've just mentioned to you, you don't have to worry about memorizing any of the little details that are there, as I mentioned to you at the outset. So this is where things begin. This is the dorsal column. So the dorsal, again, the posterior part of the spinal cord. So this information comes in, again, on this side of the body. That information goes in to the dorsal horn. That information comes into the dorsal horn because the dorsal horn of the spinal cord, the gray matter there, is where sensory information gets relayed first at the level of the spinal cord. It stays on the same side of the body. That information then its axons, this group of neurons, its axons send their projections all the way up at this level of the spinal cord in this dorsal region, and this is why it's called the dorsal region, all the way up into the level of the medulla. And it sends it up in this dorsal column here, all of the information from your hands, your feet, etc., all of the sensory information involving mechanoreceptors all gets sent up through this dorsal region of your spinal cord, all the way up to the level of the medulla. At the level of the medulla, as all of these axons now will cross over. These axons that have stayed on this one side is conveying that information now cross over to the opposite side. It undergoes what's known as decussation, and it's written for you here, that decussation at the level of the medulla. And here, even though it's the same information, it's the same tract, it switches its name from being called the dorsal column to what's known as the medial lemniscus. It's really the same pathway, it's just called different things in different regions of the CNS. And again, you don't have to worry about all of the details that are located for you here. I'm not going to talk about the cuneatus nor the gracilis nuclei. It's just to give you an idea of how things cross over. The more important thing is, as this information gets conveyed, there is an order to this information. It's not just a wild bunch of information from your feet, your hands, and it's being mixed up. There is an order to the information that's being conveyed through the mechanoreceptors all the way through to the level of the thalamus, and then from the thalamus all the way up into the level of the cortex. So here it is at the level of the thalamus. So the thalamus being part of the diencephalon, and here is where it makes a synaptic connection. So this is the third group of neurons, and this third group of neurons will then send its axonal projections up into the cortex and specific areas of the cortex. This area of the thalamus is one that I'd like you to make sure that you know. There are lots of regions in the thalamus, and this is what's known as the ventral caudal nucleus of the thalamus. Okay, so mechanoreceptive information is organized in what's known as a somatotopic way, and I'll show you what somatotopic means in a moment. All the way up, it crosses over at the medulla, gets into one area of the thalamus, the ventral caudal nucleus of the thalamus, and then from there it will go to a specific area in the somatosensory cortex. Do you have any questions about this at all? Yes. Sorry? So we'll talk about the ventral caudal and other nuclei within the thalamus. So different types of information hit different nuclei, and you'll see that in the next few slides. Pain information from the spinal thalamic hit a different nucleus in the thalamus, etc., all the way through. But most sensory information, including visual, get targeted to the thalamus, and this is one nucleus that is responsible for mechanoreceptive information. So from the thalamus, from that ventral caudal nucleus, it then sends a projection. In order for you to make any sense of what you've just been touching, the thalamus isn't going to be good enough. The thalamus can't process what you've just been touching. Only the cortex and those mini columns in the cortex allow you to be able to tell what the difference is between touching something that is metallic versus something that is plastic versus something that is furry versus something that is completely wet, etc. In order for you to be able to discriminate those different types of mechanoreceptive touch sensations, that information from the ventral caudal nucleus has to be sent into an area of the cortex known as area 3B. It's also called S1. And all of this really means that it's somatosensory. It detects what is going on in the world around it. So this ventral caudal nucleus will send information to different regions of this area of the cortex. Again, just to orient you a little bit in terms of what's going on, this is the central sulcus, which divides the different regions of the brain. So what is the region of the brain that is found here in front of, if you want to think of it that way, the central sulcus? So remember, 4 regions of the brain. What is it? So this is the frontal cortex, and here is where area 3B is. Here is where S1 is in purple. What is this area of the brain called? This is the parietal cortex, and this is the first part of the parietal cortex known as area S1, area 3B or S1. And if we look at this gyrus, this sort of outfolding, in area 3B, we'll get specific axons coming in, conveying information from the ventral caudal nucleus. And again, we won't worry too much about all of the information about the different regions surrounding it, but we'll see that For example, area 3B, it contains a complete map of your body, a somatosensory map. All of the different areas of your body are represented in this one area of the cortex. The other thing that you'll make note of, and we'll talk about this in a little bit more detail, is the information doesn't stop there. And again, you have to think back, and you should be doing this throughout this course. If you're looking at this diagram, the axons don't just stop at area 3B, right? So they've come in through the ventral caudal nucleus of the thalamus. They've gone into area 3B. They tell this area of the cortex what's going on. This area of the cortex has those mini columns that are processing all that information. But what happens to the information next? What do you see those arrows doing? So they're going to other areas of the cortex, like area 2 or area S2. So think back to that slide that I showed you earlier. So all the cortex is organized into 6 layers. Which one of those layers actually has groups of neurons that actually project to other areas of the cortex? Is it layer 1, 2, 6? So remember, at the very end, in going from one area to another, it's really the layer 6 group of neurons in this area of 3B that are sending their projections after they process information to other areas of the cortex. So again, before we get there, it's useful to go back and look at what's known as somatotopic or topographic organization of this cortex. The brain is an elegant structure. It has many, many neurons, many, many connections, but it's organized in a logical, cohesive manner. So it has 6 layers all the way through. But if we're looking at it, each of those different columns that we see, those little mini columns, if we're looking at the very middle portion, so right in the midline at the very top, all the way through to the side here at the lateral sulcus, so right on near the temporal lobe, if we looked at this little chunk of the post-central gyrus that makes up area 3B, we would see that there is an organization to how information comes in from the ventral caudal nucleus into this area 3B. So we'll have all of the information coming in from the lower extremities organized in a very logical topographic manner. So the foot is near the leg, for example, and the leg, all the somatosensory information coming in from the leg to the ventral caudal nucleus, and then from there into the cortex. Everything would be organized from the leg to this area of the cortex and the trunk, etc. The other thing that you'll notice about this is that not all of the areas are actually the same size. Does that make any sense to you? Yes? Why does that make sense to you? Okay, perfect. Almost. So some areas are more sensitive. So what do we talk about when we talk about sensitivity? We talk about receptive fields. So some areas have more receptive fields. They have more receptors packed per unit area of your skin. For example, your hand might have more, your upper lip, your lower lip, etc., might have a larger density of somatosensory receptors, and so they have a bigger representation, for example, than a structure which is actually much bigger in real life. Your foot is much bigger than your lip, for example, but in terms of the topographic organization, your lips have the highest density of these different structures, receptive fields, and as a result, this is why we have this type of topographic organization. You'll see in your hand, in particular, your fingers have a large area in the cortex that's being represented. So within each of these areas, so for example, if I looked at the area 3B that's related to the hand, we have even more organization. So not only are different chunks of the cortex organized topographically, so the foot is next to the leg, which is next to the trunk. But in addition to that, within each part of the cortex that's related to the hand, we'll also start to see these columnar organizations. I alluded to that earlier on, so 6 different layers. And here are those 6 different layers again for you. One of the things that we didn't see very much of in that previous diagram, we saw layer 6 is an output neuron that goes to other areas of the cortex. Layer 4 is actually where we get information coming in. We had looked at area 3 or layer 3, but we didn't really see layer 4. And layer 4 is something that I want you to be aware of as information coming in from the thalamus. It will make its first synaptic connections here at layer 4, and different hypercolumn neurons will respond to that information that's coming in, and what it decides to do with it in terms of sending information out, whether it's to different areas of the cortex, will be organized in this column, okay? So layer 4 is where all of that thalamic input comes in. So this hypercolumn is a block of block of the cortex that contains the neurons that are responsive to the same area of the skin, and all of those neurons in this column would respond, for example, to this sort of stimulation, and it would be detected here. And this stimulation actually would be in a different block. It would be in a totally different area of the cortex. It wouldn't be in this block of the hypercolumn that would be stimulated. And all of this is organized in a topographic manner. We know all of this, by the way, through earlier studies that were done through electrically stimulating the brain. So early on in the 1950s and 1960s at Montreal, they did electrical stimulation of the brain. They saw what sorts of activity were elicited. Nowadays, we're a little bit more sophisticated, and so we can actually look at the somatotopic organization of area S1 using what's known as fMRI, or functional magnetic resonance imaging. And using functional magnetic resonance imaging, where you go and stimulate while someone's in an MRI and you stimulate their finger, one of the things that you can see is even with this technique, we still see somatotopic organization. So if we stimulate, we'll actually be able to pick up different areas of area 3B or S1 being stimulated. And of course, never let it be said that PSL300 gives you sort of the middle finger, right? Because this is one, I don't know why they picked this particular slide, but there you have it. You've gotten the middle finger in PSL. So why do we have those area 6 neurons? What is the function of these area 6 neurons or layer 6 neurons? So if you look at it, this is area S1. And right behind area S1, we have these other slabs of the parietal cortex. So why have all of those other areas in the parietal cortex? It's relatively big. Why have all of that information sort of out there? So behind it, we have all of these association areas and the somatosociation areas that are just behind or posterior to that S1 area. And what this does is, as that information comes into the ventral caudal nucleus from the thalamus, gets processed in area S1, integrates that signal, and then it leaves area S1 saying that you've just touched something that is metallic, or something that is cold, or whatever it is, or you've just touched something or had stimulation. It will then send that information to the association areas around that area S1. And this is where, for example, you might get more complex types of stimulation. So you might be able to recognize through activity as more and more of the information gets processed in association areas that your skin is being stroked by something that is metallic versus something that is wooden, etc. So all of these association areas are really important that way. And we'll find that this is exactly the same model no matter what we look at. When we look at vision, we start off in this one small area of the cortex, and then we have all of these other association areas that are built up around understanding what is actually happening in the outside world. The one thing to make note of here before I go on, these Pacinian corpuscles, because they have really, really large receptive fields. Remember, it takes up literally about half your hand. These actually don't enter into the somatosensory cortex directly. They'll enter into an associated area known as area 1 that is right behind or posterior to S1. This is where you start processing whether you have movement, etc. But because it's such a large receptive field, it can't enter into S1, which normally picks up on smaller types of receptive fields. So the last thing I'll talk about in terms of this association cortex are these areas 2 and 5. Right behind this area, area 1, are these areas 2 and 5. And this is where you get the postural, the joint type of information being processed. These tell you, for example, when your arm is in one sort of orientation versus another orientation. It will give you some idea as to the types of stimulation that you have. And there again also is a representation of the body image. It will tell you a lot about how your arm is in space, and it will actually tell you if your arm is flexed, whether it's extended, and it will fire at different points in time depending on what's going on in the different areas of the cortex. Do you have any questions about that before I go on? No. Are you going to need time to process that information before you yes, okay. That's fine too. Okay, so we'll try to keep this simple as well. Again, I know there's a lot of information. I apologize for it. This is such a condensed group of lectures. Normally, you'd string it out over about a week or so, but it's a lot of information, and there's a lot of neuroanatomy, I know, so I apologize for that. When we talk about this other tract, we're talking about the spinal thalamic tract. This is a little bit different. So remember the dorsal medial, the dorsal column medial lemniscus pathway? Most of it was ipsilateral. I touched something on this side, it stayed on the right side of my spinal cord, it went all the way up and then crossed over at the medulla. This is different. So the spinal thalamic tract is actually really different. So when we look at this, the neuron, as the sensory information comes in from one side, it crosses over almost immediately to the opposite side of the spinal cord. It will then go up in this part of the spinal cord, not in the dorsal part of the spinal cord, but in the ventral region of the spinal cord, in that funiculus that we looked at earlier. It will go up all the way in this region of the spinal cord, all the way up to the brainstem and into the thalamus. So it crosses over almost immediately. Your pain pathways are very different than your touch pathways, in other words. And all of the information in the spinal thalamic tract involves things like pain, and this is one of the major pathways for pain. It will convey information related to touch, temperature, as well as itch. A very small amount of touch. This is not the major pathway for touch. That's the dorsal column medial lemniscus pathway, but I just put that there for completeness. We'll concentrate on pain, temperature, as well as itch. If you get asked that on a multiple choice test question, spinal thalamic pathway is really pain, as well as temperature and itch, not related at all to touch in terms of this course. Again, we won't worry about all the details here. There are different regions of the spinal thalamic tract. It's really to convey this information. The crossing over is important. You should know that it crosses over as soon as it enters into the dorsal column, the dorsal horn. It will cross over to the other side, and it will go up this region known as the anterior white commissure, and it forms, the axons form this spinal thalamic tract, just like the dorsal column did. So again, there are two different pathways that it can take up to the thalamus. So if that information is coming in from one side and it synapses at a superficial layer known as lamina 1 of the dorsal horn, then it will convey information related to pain, itch, as well as temperature. And again, it has two different pathways. There's a medial pathway that involves those C fibers, and the lateral spinal thalamic tract, which has both A delta and A beta fibers, which uses the anterior spinal thalamic tract. And again, we'll talk a little bit more about that later on in the course. I just wanted to introduce those ideas to you, that there are different modalities for pain being transmitted up to the thalamus. You don't have to worry about the details that are shown for you here. It kind of outlines the different paths that these take. It's more important to concentrate on what I just highlighted for you here in the actual text on the right. One of your colleagues asked, if we were to take a look at the thalamus, and this is the thalamus sort of cut in cross-section from the side, it's a sagittal section. Unlike the ventrocaudal nucleus that was mechanoreceptive that would be found up here, the spinal thalamic tract would actually make connections primarily into another area of the thalamus known as the ventral posterior lateral region of the thalamus. It's a different nucleus. It's a different synaptic connection than you have for the mechanoreceptive type of information. I want you to be very clear on that, especially on the midterm, how this information comes in, where it makes synaptic connections into the thalamus, okay? So pain and temperature information and itch information comes here into the VPL. Mechanoreceptive somatosensory information goes into the ventral caudal nucleus of the thalamus. Very, very different. And you need to be very clear on this on the midterm. I told you earlier on that in the spinal thalamic tract, one of the types of neurons that we see a lot of are these wide dynamic range neurons. And these wide dynamic range neurons signal through this spinal thalamic tract. And these wide dynamic range neurons exist because multiple types of information make synapses on to this type of cell that's found in the spinal thalamic tract system. So these neurons respond both to light touch as well as to intense stimuli like pinch. They will also respond to things like heat as well as to cold. So in other words, they have input coming in from mechanoreceptors. So these are mechanoreceptors that signal through the A beta fibers, and they signal through the nociceptors, things like pinch as a nociception, heat as well as through cold that signal through A delta and C fibers. Both of these come in, make synapses onto this wide dynamic range cell, and this wide dynamic range cell will send its axons up through the spinal thalamic tract. And I'll get back to that again in a few slides. This is part of the spinal thalamic tract, and I wanted you to be aware of it. The last type that we're going to be looking at is this spinal reticulothalamic tract. Again, their projection neurons are a little different. They are in this intermediate zone that you would find here. They send their information bilaterally, both sides, not just on one side. They send it bilaterally up both pathways, both parts of the spinal cord. They have very different types of input. They have highly convergent types of input. They will signal to a different area of the thalamus. So they will send information to the central median nucleus of the thalamus, and the spinal reticulothalamic tract, the spinal thalamic tract, and the dorsal column medial lemniscus tract all have different targets in the thalamus, all convey different types of information, as we'll see in a moment. So these, again, are really pain and the slow pain pathway that is sending information up into this type of tract. Do you remember what type of axons we would find with this slow pain? So the C, which is what? So it's unmyelinated, small diameter. So in this pathway, we would only find small diameter, unmyelinated axons that are going up bilaterally in this area of the spinal cord. So it sends information in this area of the spinal cord, as you can see outlined. Eventually, it will make synaptic connections up here in the central median nucleus of the thalamus. So I'm going to switch gears just a little bit and talk to you about pain. So I'm going to talk to you about two different aspects of pain. We saw that there are areas that have convergent input, right? So I'll talk about that, and I'll talk to you about when pain signaling goes a little bit wrong. One of the things that you might experience at some point in your life is what's known as referred pain, and it's a physiological response due to the fact that different nociceptors actually converge on to the spinal thalamic, and in some cases spinal reticulothalamic. These are those wide dynamic range neurons that I mentioned to you earlier, and these projection neurons receive input not from just one place in the body, but from multiple places in the body. What does that actually mean? So, for example, you might have two different pain signals that normally come from your skin. That's, again, part of your somatosensory system. It responds to signals on the surface of your skin. Might be a patch of skin at the back of your body. But you also have coming in to this spinal thalamic and maybe spinal reticulothalamic tract, you might also have nociceptors that are embedded within an internal organ. So you might have nociceptors that are associated with the kidney, for example. But what does that feel like? What does it feel like when your kidney is actually having pain? Nothing. what it feels like is that it feels like a patch of skin on another part of your body is actually in pain. So you might have a kidney problem where your kidney is malfunctioning and it's signaling that something is going wrong, that it's inflamed, there's an infection, that it is undergoing some sort of inflammatory response. It's signaling through its nociceptors, but your body doesn't know that. How it responds is you might suddenly have a pain in your back. And you might be thinking, wow, this is really weird. I have a pain in my back. It's a referred pain. This is what referred pain is. It's being referred to a different part of your body. And so as a result, this visceral pain gets miscommunicated as a patch of skin that is being damaged somehow. And that's the way that you experience a visceral pain, something with an internal organ, as a patch of skin being hurt, even though you've never hurt that part of your body. And it's because these two different types of sensory information are converging on that same wide dynamic range neuron that makes up that spinal thalamic pathway. The more common way to think about this is this pathway. And there's a lot on this slide, and this is sort of the last slide I have for you, and then we'll sort of wrap things up. So this is an important slide for a variety of reasons. This is that same wide dynamic range neuron where we might have, for example, two different inputs coming in and working on that wide dynamic range neuron. I told you at the very outset that you might have mechanoreceptors as well as nociceptors all making synaptic inputs onto this spinal thalamic wide dynamic range neuron. So these large mechanoreceptors, for example, could come on and through interneurons that are found in this region of the dorsal horn presynaptically inhibit these nociceptors that are also coming in as well. So we have these mechanoreceptors coming in, the A betas, mechanoreceptors, and then we have the pain pathway, the C fibers that come in that signal for pain. And both of these are synapsing onto the same neuron. Everyone agree on that? You all see that? So we have mechanoreceptive input coming into this wide dynamic range neuron, and we have this C pain neuron coming in, also synapsing on this. We also have these interneurons that can inhibit this pathway. So these inhibitory neurons that are found in this layer known as the substantia gelatinosa, which is denoted here as SG, are found in the spine, in the dorsal horn. So imagine this, right? So why is that important? So imagine that you've been working, you've been building something, and you've hit your thumb with your hammer. That is a type of mechanical damage that is done to your thumb. You'll feel that pain. It is being transmitted to your spinal cord via the C fibers, right? Yes? No? So what do you do when you hit your thumb with a hammer? You rub it. Okay. That's one thing that you could do. You could rub it. What else could you do? You could cry. Yes. Okay. Also a very good response. In other words, you mechanically stimulate it. And why does that? Why do you do that? It's because in theory, as you are mechanically stimulating the same finger that you've damaged, or you could stick it in your mouth and suck on your thumb if it hurts, what you're doing is you are activating this A beta fiber, the mechanoreceptors in your thumb that you've just hit. You're feeling the pain, and by sucking on your thumb, by massaging your thumb, by activating the mechanoreceptors, that signal is now coming in. It is causing these interneurons to be activated, and it will turn off a little bit the pain signals that are being integrated at the level of this wide dynamic range neuron. So this is also why, for example, if you are feeling like your shoulders are achy, why having a massage on your shoulders actually feels better, or it makes your shoulders feel less achy. The massage is mechanically stimulating the same mechanoreceptors that are signaling for pain. And again, we won't get into all of the other sort of wide dynamic range neuron activities, but I did want to make that relevant for you. The last thing I'll mention to you is they will also send projections up through this track known as Lissauer's track, and these projections from the wide dynamic range neurons can actually go up multiple spinal cord levels. I know some of you have to get to tutorial. If you need me to, I will post this for you somewhere, but this is another example of a question that you might be asked. Again, if you need to see me for tutorials or extra help, please let me know, and I'll see you all on Thursday. So if you have any questions, I'm here early. Ask questions during the time that I'm setting up.